All right. So thank you all so much for joining us today, and good afternoon. I'm Dr. Lee Frame, and I'm honored to be your host today. I serve as the Executive Director of the Office of Integrative Medicine and Health, the Director of the Resiliency and Wellbeing Center, and Chief Wellness Officer for GW Medicine, and Associate Professor here at the George Washington University School of Medicine and Health Sciences. On behalf of our entire team, I'm thrilled to welcome you to the 8th Annual Sung Symposium, hosted by the Office of Integrative Medicine and Health. This year's theme, Fasting for Wellbeing, invites us to explore one of the most ancient yet increasingly evidence-based tools in the health and healing toolbox. From time-restricted eating to longer fasts, and from cellular repair to spiritual renewal, fasting spans physiology, psychology, and tradition. Today, we'll hear from leading experts who will illuminate the science and practical implications of fasting across different contexts, populations, and clinical conditions. Before we begin, I'd like to share a bit of background on integrative medicine and its history here at GW. So what is integrative medicine? Integrative medicine and health reaffirms the importance of the relationship between practitioner and patient, focuses on the whole person, is informed by evidence, and draws on all appropriate therapeutic and lifestyle approaches, healthcare professionals, and disciplines to achieve optimal health and healing. It is not about choosing between conventional or complementary. It is about integrating the best of both to support the whole person. GW's integrative medicine journey began in 1998 with the founding of the GW Center for Integrative Medicine by Dr. John Pan. Since then, it has served the Washington, D.C. metro area as a beacon of whole person care, including a training site for the GW MD program and for our fellowships in integrative medicine and integrative geriatrics. In 2015, we expanded our educational offerings with the launch of graduate programs in integrative medicine, designed to equip practicing clinicians with the tools and knowledge to provide truly patient-centered integrative care. The Office of Integrative Medicine and Health was formally established in 2017 under the Department of Clinical Research and Leadership. In 2018, we launched the GW Integrative Medicine podcast, which you can find on our website or wherever you get your podcasts. In 2019, we introduced our integrative nutrition programs, further strengthening our commitment to evidence-based approaches at the intersection of food, lifestyle, and health. While these graduate programs are no longer accepting new students and are now in a 2-year teach-out phase to support our final cohort through graduation, their legacy continues. The office remains committed to the educational mission we began through these programs. As these programs sunset, the role of the Office of Integrative Medicine and Health becomes even more critical. Our operations depend on philanthropic support, and now more than ever, donations are essential to sustaining our mission. Continued support allows us to maintain key initiatives, including our institutional membership in the Academic Consortium for Integrative Medicine and Health, a resource that ensures access to evidence-based integrative health education for the entire GW community, and this free annual symposium. Since its inception, this annual symposium has embodied our mission to educate, inspire, and bring together experts, learners, and practitioners from across the country and increasingly the world. Past topics have included mental health, resilience, the gut and its microbiome, and last year, sleep for well-being. All past symposia are freely available on our website and YouTube channel as part of our commitment to open, accessible education. And now in 2025, we turn to fasting. From ancient religious rituals to cutting-edge metabolic research, fasting challenges our assumptions about what it means to nourish and sustain the human body. It's not just about abstinence, it's about rhythm, repair, and resilience. Thank you for joining us today. I look forward to a meaningful and thought-provoking afternoon together. Let's begin, and I'm going to turn it over to our namesake, Dr. Pat Sung. Dr. Pat and his lovely wife, Marguerite, gave the seed funding to found the Office of Integrative Medicine and Health, which is why the symposium is named after them. Pat, I know you want to say a few words.
Hi, everybody. Pat Sung here. Welcome to our seminar online. Well, I'm here in Hong Kong with my wife. It's 1 a.m. here, so I won't stay long. So congratulations and wish you guys have a great seminar. Intermittent fasting is, I'm very interested in it, so I'm really looking forward to this.
Thank you so much for joining us. What commitment. I know you are going to pop out and catch the recording because you need to get your restorative sleep in, right? That's very important.
Yes. I need my rest.
Well, thank you. We appreciate it.
Okay.
Next on our agenda, I am thrilled to turn it over to the current medical director of the GW Center for Integrative Medicine, Dr. Mikhail Kogan.
Well, thank you. Thank you, Lee. Thank you, Dr. Sung, for making this happen and donating the seat to get this going. So I have an immense pleasure to introduce our first speaker today. Valter Longo is one of the world's leading experts on gerontology, and so I'm quite excited since my main field is geriatrics. Every time I get to hear him talk, it's always something new and exciting. He is the outmost expert on studying the fast mimicking diet, also assessing chronologic age, and not just chronologic age, but rather functioning age, particularly on cellular level and tissue level. He's Edna Jones Professor of Gerontology and Biological Science and Director of the Longevity Institute in the University of Southern California. His laboratory has identified some key genetic pathways to regulate aging. I'm sure he's going to talk about this today quite a bit. I understand that Valter is currently in Italy, so I hope that the technology will be our friend today. They'll be able to hear everything well, but that highlights the fact that he is traveling all the time. I'm sure he's going to talk about some of the Italian longevity. Of course, the part of Italy, Sardinia is in blue zones, so we're probably going to hear some of them about that. I am personally super excited, and I'm a little guilty. I haven't read his new book, Fasting Cancer: How Fasting and Nutri-Technology are creating a revolution in cancer prevention and treatment. It just came out a few months ago, so I'm guilty as charged. I will be reading it soon, but I can't recommend enough his prior book that was in 2019, The Longevity Diet, one of the books that I give to a lot of my patients. Valter, thank you so much. We are so excited to have you with us, and we'll hand it to you.
Thank you, Dr. Kogan. Thank you for the invitation. And I have to say that I was asked to do a talk on cancer, right? So, so I'm not going to mention a lot of the things that you mentioned, but I'll focus on the cancer part. And so, we've done maybe about half of my work is on cancer, so I like to focus on that. And this, I always show this slide as one of my initial slides, and this is actually the lung cancer part on the left is from a group in Switzerland, and the part on the right is our work, and this is showing cancer-free survival, right? So if you just treat mice with chemotherapy or fasting cycles, not much happens in terms of cancer-free survival. You get a little bit in a breast cancer model where we inject a few low-level cancer cells, but not much. And then you combine the two, and you combine the two, and now you see, at least in this model, it doesn't always happen like this, but in these models, chemotherapy, which doesn't cure much, now it's starting to be very effective in sort of searching and destroying cancer cells wherever they are, including in some metastatic models. So, so then, of course, we're very interested in the mechanisms on one side, but then using the mechanisms to actually come up with clinical trials that can test our hypothesis and the hypothesis that fasting and fasting mimicking diets can enhance cancer treatment. And so first of all, what are fasting mimicking diet? This started with a cancer clinical trial at Norris maybe 15 years ago, at Norris Cancer Center in Los Angeles. And the patients were placed on water-only fasting plus chemotherapy. And the patients did not want to do water-only fasting because it was just too much, too extreme. And the oncologists were not very comfortable with it. So from there we were funded by the NCI, the NIA, to develop a fasting mimicking diet. So it was developed for cancer patients. And so that's we've been almost exclusively both in mice and people, and in the clinical trial, we're using this fasting mimicking diet, which is a low calorie, low sugar, low protein, high fat, vegan composition. Okay, so then, so we've been publishing this for a long time, and one of the early studies, Cheng and Lee then in the lab showed that whereas in normal cells, AKT, TOR signaling, translation, these are all decreased during fasting. They're part of fasting responses, right? To turn down these signal transduction pathways and also protein synthesis. But in 41 and other cancers, in triple negative breast cancer and other cancer, we saw that in fact AKT, TOR, cis kinase was upregulated, right? So we kind of put it away, and then a few years ago we started working on this again, and started working on the idea that maybe there were escape pathways, right? So you starve cancer cells and normal cells are going the right direction. They know what to do, and this is a very ancient program, a fasting response program. But cancer cells are confused by this environment. Now you're reducing glucose, you're reducing growth factors, you're revolutionizing their environment. So they become desperate and they start increasing instead of decreasing pathway, right? So, and one of them was the TOR cis kinase pathway. So increase instead of decrease. So then Giulia Salvadori in the lab, and so I have two labs, one here in Milan, mostly focused on cancer. So Giulia then started looking at RNA-seq and basically looking at cancer cells and trying to figure out, this old observation that we made. Well, let's do it, looking at every pathway, right? So and so we fast the mice, we fast the mice with the cancer. And what happens in the cancer cells? How do they rewire, right? So do they do the same as we had shown a long time ago? And what are the pathways? And sure enough, Giulia then, by this RNA-seq, shows that PI3 kinase, AKT pathways, and TOR are in fact upregulated in these triple negative breast cancer cells. And so now, because TOR and PI3 kinase and AKT, now she targets those pathways with 3 different drugs in the hope that this would be enough to cause the regression of the tumor, right? So here's the tumor growth, the triple negative breast cancer. And now she intervenes with fasting plus the 3 drugs. And sure enough, right? So the cancer comes down. But the cancer then adapts and is able to grow, right? Unless there was a pre-treatment with the fasting mimicking diet. And so eventually we figured out why. And the reason was that if she pre-treated with the FMD and then treated with the 3 drugs, now the cancer comes down and stays down. And the reason for this is cancer stem cells, right? So then the cancer stem cells, so the fasting mimicking diet is narrowing this pathways essential for cancer survival, right? And so it's narrowing this redundancy and is now making in indifferentiated cancer cells, is making them rely on PI3 kinase, AKT, TOR, CDK4/6, and so a few pathways. But even though this is very effective with these differentiated cancer cells, it doesn't do much to the cancer stem cells. Now, the cancer stem cells have a different program. They're worried about sugar, right? These cells don't care about sugar. These cells are very dependent on sugar. And so now the fasting is lowering the sugar, and it's inactivating PKA, and it's also turning down the stemness genes that allow the cancer stem cells to proliferate. And so that's why we see, and actually in this paper, Giulia actually demonstrated this, but this is why we see the cancer staying down, because the cancer stem cells eventually will give rise to new differentiated cells. And so the cancer keeps coming back unless in a glucose inhibition dependent manner, this problem is solved. Okay, so what if we started the other way, right? So what if we start with drugs that block in this case, hormone therapy, these are ER positive breast cancer cells. What if we start with fulvestrant, blocking estrogen signaling, and palbociclib, blocking cyclin, so CDK4/6 pathways, right? So if you do that in these tumors, it works just like in women that receive this for a while, and then the cancer, this green right here, the cancer starts adapting and eventually starts growing. And sure enough, in 1 or 2 years, in cancer patients, this particular standard of care dual intervention doesn't work anymore. But when Irene started combined it with the fasting mimicking diet, now you see that it keeps coming down, and then you start seeing mice becoming cancer-free, right? So now, I mean, if you turn around, if we had started with the fasting mimicking diet, we will have concluded that CDK4/6 and estrogen receptor signaling was the escape pathway, right? So that's how we did it. We started with the standard of care and then use the FMD to block these escape pathways, to block additional pathways, escape pathways, and this sure enough works. And also it works in cancer cells that are already adapted and fast growing. So if we intervene now with the 2 drugs plus the FMD, now we see a reversal of the resistance acquisition, right? So then it's remarkable that Irene showed that so this is black and CDK4/6 is black, right? So the fasting mimicking diet was acting on 3 different things: leptin, insulin, and IGF-1. And if any of this was restored, the cancer will start growing again, right? So this is a really important point, because it says that if we did this for every patient, we will have to figure out all these different pathways that are important, because even between a cancer and a next generation cancer in the same patients, you could have different escape pathways. So now it would take 3 drugs to lower IGF-1, lower insulin, lower leptin, plus the 2 existing drugs. So it'd be 5 drugs required to get the same effect as we're getting now by adding a little bit. You can think about it as a wild card, right? So the FMD is making so many changes that then the 2 drugs alone are sufficient to do the job. Okay. So then Myra in the lab instead started thinking about, okay, so what is the problem with a lot of cancer therapy is the toxicity of the cancer. Even immunotherapy is very toxic, especially when you have multiple drugs being used. And so is it possible to do a very low toxicity or non-toxic therapy? And in this case, Myra started looking at vitamin C, right? So Luke Kentley, people had been testing vitamin C for decades, not very successful. And then Luke Kentley said, well, maybe it wasn't very successful because they were looking at the wrong type of cells. So he looked at KRAS and BRAF mutated cancers and showed that vitamin C treatment was in fact very toxic to these KRAS and BRAF mutated cancers. And so Myra in my lab confirmed this, right? So this is colorectal cancer, 3 different lines. So vitamin C is increasing the toxicity to these colorectal cancer cells. Doesn't do much to lung, more different lung cancer line, and a lot to the pancreatic cancer line. But now when she combines it with the fasting conditions, now you see clear synergistic effect, right? Now, again, like I showed you earlier between fasting and chemo, now we see it for fasting and vitamin C. And that's good news, because actually both fasting, mimicking diet, and vitamin C are anti-aging interventions, right? So the opposite of what chemotherapy is, right? So they're both supposed to protect an individual. And so now the possibility is that you can begin to test non-toxic intervention to treat cancer versus the toxic ones. And so this is in vivo showing HCT 116 in vivo model. You see 3 cycles of the fasting mimicking diet are able to, together with vitamin C, are able to control tumor growth. They don't completely stop it, but they certainly have the biggest effect. And as we've seen for chemotherapy, the cycles of FMD and the cycle of vitamin C are about equivalent, right? But when you combine it, then now you see a much stronger effect. And so yeah, I don't have time to go through the entire mechanism, but essentially it seems to be working like this, that normally vitamin C, as Luke Kentley had shown before, is causing the generation of hydrogen peroxide. But this hydrogen peroxide, it needs iron, free iron, to perform phantom chemistry and generate a lot of hydroxyl radical. And the hydroxyl radical is what's really doing the damage to the DNA. It's that's where the toxicity is coming from. But heme oxygenase, which is and that's what we've shown. We've shown that in response to vitamin C, the cells activate this heme oxygenase, and heme oxygenase 1 is increasing ferritin levels, and the ferritin are basically scavenging iron, the free iron. So now they're removing this free iron availability, and so now the vitamin C is toxic, but not very toxic, right? So the fasting conditions now lower heme oxygenase 1, lower ferritin, make iron available, and now the phantom chemistry can go on, and it can go on specifically in the cancer cells, because as I'm going to show you in a second, heme oxygenase 1 is differentially regulated in normal and cancer cells. It goes up in normal cells in response to fasting, and it goes down in cancer cells. Okay. And so now we were seeing the fasting mimicking diets being effective in both immunocompetent and immunodeficient mouse model, and it didn't make many sense. And a lot of our colleagues were saying, "How can it be, right? How can you see this working with or without the immune system?" And so Stefano in the lab, the Bials in the lab, started working on this and looking at both breast cancer and melanoma, and showing basically this is the breast cancer model, that only in the combination of fasting mimicking diet cycles and chemotherapy do we see infiltration, T-cell infiltration into the tumor, right? So you don't see it with fasting alone, fasting cycles alone, and you don't see it with chemotherapy cycles alone, but you see it with both. And so this was true for breast cancer. It's also true for melanoma. And so connecting with what I just showed you for colorectal cancer, the heme oxygenase becomes a key player here too. And this is what I was saying earlier. During fasting in the liver and the muscle mice, the heme oxygenase 1 will be elevated or greatly elevated. But in the tumor cells, now you see the opposite, right? So the fasting condition are decreasing heme oxygenase. So in this case, it was enough during fasting to increase heme oxygenase to then restore the ability of the tumor to grow. And okay, so then of course, the interesting thing was, well, what about if you combine it with immunotherapy? And so we had been working for a while on this, and then we got scooped by a group in Spain, and they combined it for lung cancer models, different lung cancer models, and they were able to show that in fact fasting conditions plus PD-1 checkpoint blockade was able to had very strong effects against lung cancer models. So Salvatore in my lab more recently then decided to combine PD-L1 inhibition with OX40, and OX40 is an activator of T-cell dependent tumor attack. So PD-L1 removing the checkpoint, and then increasing or activating OX40, and therefore pushing the T cells to attack the tumor. So the two are being shown to be more powerful together than each one alone. And so interestingly, the immunotherapy, and this we've seen in many models that we use, the immunotherapy does very little to the cancer progression. This is again 41 triple negative breast cancer. What works is either FMD plus OX40 or FMD plus OX40 plus PD-L1 inhibitors. And so why does it work? Well, Salvatore did single-cell RNA-seq, and in fact showed that compared to the immunotherapy, to the two immunotherapy drugs alone, which is here in lane 2, the FMD plus the immunotherapy drugs cause a major activation in at least 3 different populations: activated CD8 cells, TCF7 positive T cells, and CD4 T cells, right? So these groups are undergoing a major expansion. And Salvatore also showed at the single-cell RNA-seq level that, in fact, the number of activated CD8 cells was flat until the FMD and the 2 checkpoint inhibitors were combined. And this is just showing the expression, the gene expression typical of activated T cells. So this is showing that going to a different triple negative breast cancer model, showing that this is not about 41s, and this is so Salvatore used this TSA, and again now working even better. Now the combination of FMD, OX40, and PD-L1 is flattening the cancer growth. Okay, so another thing that I mentioned earlier is the toxicity, right, of immunotherapy, chemotherapy, and lots of other therapies. So interestingly, we started seeing in the 41 model, when we use either PD-L1 inhibitors, OX40 agonist, or PD-L1 OX40, all the mice died, right? So and what happened is this major activation. This is OX40 PD-L1. You see major activation of eosinophils, major activation of basophils, or not activation, but increase in number of basophils, and immature granulocytes. And in all cases, you see that if you add the FMD back down to normal in all 3 different populations, right? So this may explain why the FMD plus the checkpoint inhibitor now is protecting, not only is causing a much improved killing of the cancer cells, but it's now preventing this very clear death of the toxicity of the therapy, probably caused by this activation. Okay. So then the last model I wanted to show you is a blood model. This is acute lymphoblastic leukemia, ALL. And Roberta in the lab look at the standard vincristine treatment, so chemotherapy alone, fasting mimicking diet cycles alone, chemotherapy alone, or the 2, right? So the 2 combined, again, not only improve survival, but long-term survival, like cancer-free survival, right? So now get about 60% of these ALL mice to be cancer-free long-term. And so why is this? Well, Roberta showed that the fasting mimicking diet conditions are now inhibiting lots of autophagy markers, particularly in combination with vincristine, right? So now the autophagy markers are down, and this autophagy is probably at the center of this increase in apoptosis, again, only in the combination of fasting plus the chemotherapy. And so then Roberta looked at what if I just use an inhibitor of autophagy and use chloroquine, and sure enough, the combination of chloroquine plus the vincristine, so chemotherapy plus autophagy inhibitor, was sufficient to get this cancer-free survival, long-term cancer-free survival. And again, she showed that if we use an anti-CD8, so if we remove immune T cell function, that effect goes away, and so we are now the cancer starts growing again, and it reverses the effect of chemo plus vincristine. I mean, chemo plus FMD on cancer progression. Okay, so this is just the summary of that. So normally fasting causes autophagy, increased autophagy. Here again, it turns it around, right? So now fasting is decreasing autophagy, and this decrease in autophagy together with the chemotherapy is essential to promote cell death, and it promotes cell death in a immune-dependent manner. Now, probably we're still looking at the mechanism. Why are the two things working either with or without the T cell presence? And probably, although it works better with the T cell presence, probably because a lot of these mechanisms are free radical dependent. So hydroxyl radical, superoxide, hydrogen peroxide. So then the damage is done in the cancer cell by the free radical, but this is probably also making it much easier for the T cells to recognize the cancer cell, right? So this is our hypothesis that is a cell autonomous damage attracting immune infiltration. Okay, so and this is just the whole thing sort of put together. Where the fasting or fasting mimicking diet is really going after lots of different systems. So it's lowering IGF-1, it's lowering glucose, it's lowering insulin, it's forcing the cells to go back to a mitochondrial-dependent oxidative phosphorylation, even when the cancer cell can no longer use mitochondria because mitochondria may have been damaged, or the genes that are involved in mitochondrial respiration are damaged. And so and often we see this burst of reactive oxygen species, and now this ROS can do damage to the nucleus and to the cell in general. And again, now we think that all this intracellular damage now is making it potentially is affecting T regulatory cells, and that's also something we see, a reduction in Treg. But either in a Treg dependent manner or Treg independent manner, is allowing the T cells, the cytotoxic T cells, to attack and continue the destruction of cancer cells. Now, there's now also a very interesting macrophage NK cell story. Now, this immune world is very complicated. So now we're looking at every cancer, every immune cell type, to see what else is involved and why. Okay, so what about clinical trials? Well, there's maybe 20 trials now that have been completed with fasting, mimicking diet, or fasting and cancer. One of the largest one was this one done in Leiden, 125 patient, HER2 negative, stage 2-3 breast cancer. And so in this trial, the group at Leiden saw a major increase in the non-responsive population. If we look at imaging, so clinical response, the patients on a regular diet were much more likely to not respond to the chemotherapy. And these were chemotherapy cycles given before the tumor were removed from the patient. And now, if you break it down based on the ones that were compliant to the receiving the fasting mimicking diet cycles versus the one that were compliant to the regular diet, the control diet, now you see a 5-fold difference in the portion non-responding. Now this is we just looked at the data. It doesn't look like this is very impressive, and I'll show you in a second now. This will be confirmed with triple negative breast cancer metastatic, but this is probably not the stage where FMD is going to be most effective. Why? Because here's before surgery, the patient just do a few cycles of the fasting mimicking diet, and then that's it until, you know, the cancer may progress and patients go on to do something completely different, right? So this was showing fundamentally what we see in mice is happening here, but again, it doesn't look like this is probably going to be the just a few cycles of FMD are not going to make that big of a difference in overall survival. Okay, but the Vernieri group instead is looked at lots of cancers as well as the Nencioni group in Genova. And in this paper published a few years ago, they saw evidence for the fasting mimicking diet cycles reshaping systemic and intratumor immunity, right? So this is good news, and it's probably the same infiltration that we showed for the mouse studies they saw in patients. And this seems to be very consistent. So that's good news, suggesting that maybe that free radical dependent damage that we saw in the mouse cells in lots of different mouse models, now they're also occurring in people, and the T cells recognize that damage and attack it. I mean, I'm speculating here, but certainly, so the evidence is there. Now, whether that's a mechanism or not, we don't know yet. Okay, another interesting thing that they published recently was out of that trial that I just showed you, they had, I think, about 20 stage 4 metastatic cases. And out of the 20, 5 of them showed what they call exceptional tumor responses, right? So stage 4 metastatic type of cancers that you don't usually see going into remission. But here, these 5 cases, I think it was colorectal, breast, pancreatic, and lung. And all of them went into remission in a very surprising way. So again, you know, anecdotal, sure, but very interesting. And again, matching the still preliminary but long-term data. Here is triple negative breast cancer. So this is 41, and the triple negative breast cancer either treated with chemotherapy alone or chemotherapy plus the fasting mimicking diet cycles. So now this is a much more promising way to use the FMD long-term together with the chemotherapy, not just a few cycles, let's say very early on before surgery, and then, you know, hoping that this is going to do wonders, 5 years later. So yeah, so now we're seeing Ligorio and Vernieri saw a nearly doubling of overall survival after almost 4 years. So some of the concerns have had to do with muscle loss. And so here in Genova, they looked at the 8 cycles of the fasting mimicking diet and showing that in patients, in women that were doing this, there was no decrease in hand grip strength. In fact, if anything, there was a potential increase in muscle function and a potential increase in lean body mass, not decrease, but increase. And then this is very consistent with what we see in the diabetes trials, in the pre-diabetes trials, and lots of all the trials that we ran. The fasting mimicking diet has a short-term effect on muscle mass, but then long-term, the absolute mass stayed the same. The relative mass, lean body mass, actually increases. Okay, and then this was just published by the Vernieri group, and showing again, well, actually mechanistically showing the early down modulation of tumor glycolysis was predicting whether the triple negative breast cancer patients were going to be sensitive to the fasting mimicking diet or not. But the really nice thing is that, and this was FMD plus chemo plus or minus metformin, right? And the metformin made no difference. So all the major job was done by the FMD, and this nearly doubled or had a major increase in the complete pathological response, and nearly doubled the complete pathological response. So this is a surrogate for overall survival. And so matching the other papers that the Vernieri group had published. Okay, so I'm just gonna end there. And since I cannot join you later, if anybody has questions, you can please ask me. Thank you.
Great, thank you so much. We definitely do have some questions. I think that a little refresher on what makes the fasting mimicking diet unique in terms of its formulation would be helpful. And how does that compare to, say, fasting? How is it different than fasting in terms of, what you are eating or are not eating?
Yeah, I always say that fasting really doesn't mean anything. It's like saying eating, right? Well, is eating good for cancer? Well, yeah, it depends
Right.
on, lots of details, right? Yeah, so I mean, the fasting making diet is a way to try to bring the standardization. I mean, first get the effects of fasting, and then standardize it, and then protect the patient from hypoglycemia, from the hypotension, and lots of problems that can occur. I mean, most physicians will say don't do fasting at home, not long-term fasting, and so the fasting mean diet is standardized that and made it so that people can do it at home. Now, in formal trials, it's been tested probably on over 1,000 cancer patients. So, so far we really don't see a problem with it. And yeah, so I think again, that's how it got started, right? It got started with water-only fasting and patients refusing water-only fasting. And then, we moved to the FMD. Well, we moved to developing the FMD and then testing only the FMD. And now all the hospitals that come to us for, so I think there are now 20 trials or more ongoing. And I think it would be very difficult to get these hospitals to do water-only fasting trials, right? But they're all excited about the FMD type of trial because, it comes in a box, it's given to the patient, and it's pretty straightforward for the patient. And the patients, in most trials, there has been high compliance.
Mhm. I like to think of the fasting mimicking diet as a diet that both feeds your gut microbiome, your gut microbes, with fiber, and then also feeds your brain with healthy fats. That's my impression of it. I think there's a little bit of confusion as to, can you do it? Does it have to be vegan? Can you do this with animal protein? One of the questions in the chat.
I don't think so because of amino acid composition, right? So we're really targeting lots of the amino acids that are very low in the vegan diet, in the vegan proteins, so methionine, leucine, et cetera, et cetera. Yeah, so I would say, and sometimes the difference is over fivefold, right? So this is not a 10% difference between an animal protein and, let's say, a legume protein. It's over fivefold. And so you can see how now, if you increase the level of an amino acid by fivefold, this could interfere with everything we're showing you.
I have a question for myself. One of the things I've been thinking out, and I haven't seen it specifically studied, but I feel like you probably know the answer to this. One of the things that is more popular in athletes and high-end users of supplements is the use of amino acid supplements. And how can amino acid supplements affect this pathway? Obviously, it's not what you're typically studying, but right? Like you're talking about methionine is something that's really going to activate mTOR, and that is going to potentially undo some of these benefits.
Yeah, no, we studied the amino acids, right? So we're very, in fact, we were about to publish several papers on amino acid. And so, yeah, for cancer, this is particularly problematic, right? Because lots of cancers will survive if you give it the right amino acid, right? And so they thrive on, let's say, glutamine, and there is a few amino acids that the cancers love, and love in a metabolic sense, meaning they can be a precursor of lots of metabolites that they need to stay alive, right? And yeah, so then for a cancer patient, it's a bad idea. For a normal person, it depends, right? If you're malnourished, it's very good, right? If you don't have that amino acid, then it's starting to affect your immune function, your muscle mass, etc. Yeah, then you need it. Most people are not malnourished. Most people have too much proteins. And, and so these amino acids are at the center of increasing growth factors and increasing anti-apoptotic factors. And so, yeah, so this is why the mice that, and the people, but the mice that have the record longevity extension have a mutation either in the growth hormone or in the growth hormone receptor. And so this is the central amino acid pathway, if you want to call it that, right? So blocking the amino acid response pathway is causing this 40% lifespan increase. And then interestingly, if you take the mouse and you calorie restrict it, you further cut the amino acids in the diet, then they go to, this is work by Andrew Barkey, they now live twice as long, which is really remarkable, right? To make a mouse live twice as long.
I think Dr. Kogan,
Yeah,
I know
so-
Dr. Kogan's going to have questions.
Well, yeah, so my question is obviously going to be a bit more clinical. So, Walter, I mean, I think it sounds like we getting close to say there is enough high quality evidence to say maybe it's time to start talking to the patients about implementing this on a broader scheme, especially since we know the safety here is really good. In the worst case scenario, well, we may not be necessarily benefiting every cancer, but we definitely going to be safe. And in clinical realm, people seeing your work and weird patients asking, should I be doing this? And of course, you can't really answer every clinical situation, but I want to get your general take on things.
Yeah, I would say for diabetes, prediabetes, this is much more advanced, right? Much simpler. So we now have done 5 trials and there's more coming and very consistent effects, right? So I will argue that there is a competition for GLP-1 that is much safer, much more going after the problem and not putting a Band-Aid on the problem, like much more real treatment than management, right? So now, a lot of these drugs are managing the disease. For cancer, I think that it's more complicated, and it depends. It's 100 cancers, 1,000 therapies. So I think for sure, what we usually say to oncologists is, if you're running out or you're about to run out of options, then talk to us, right? Because now we can strategize and we can say, well, this is not like, I just presented, I say hormone therapy. Well, it works, and then the patient becomes resistant. Let's say now the patient became resistant with fulvestrant and palbociclib or ribociclib. Okay, now talk to us, right? Because you may be in trouble, right? So what do you do next? Well, okay, you could try things, but yeah, so there's not too many viable options. So at that moment, I think it's good to start thinking about the fasting mimicking diet. In early cases, like I showed earlier, stage 1, undergoing surgery, 96% survival, I would say let's wait, right? Because what if this is making things worse, for example, right? I don't know, but it's certainly always possible.
Well, I think that's very exciting. I know it's been a while since we've had some major breakthroughs in cancer. We got the immunotherapies, which are fantastic, but they only work for certain cancers at this point. And it seems like perhaps they could work even better if we could put them with this adjuvant therapy, which has pretty low risk. So I'm personally very excited about this. I hope that this will be something that in the future we'll be able to know exactly who's going to benefit from this and start using it with whatever the best combination therapy is from the get-go and have better outcomes.
Yeah.
Any other questions? Well, we have Dr. Longo. Well, as a reminder, Dr. Longo was here at the Sung Symposium 4 years ago as well and gave a nice introductory talk to fasting mimic diet, metabolism, and longevity. So you should check that out if you haven't watched it yet. I know Janet put the link in the chat, and I'm sure she will be including it in follow-up materials as well. But with that, I will let you go, Dr. Longo. Thank you so much for joining us.
Okay, thank you. Thank you. Bye.
All right. And now I have the honor of introducing our next speaker, Dr. Courtney Peterson, PhD, Dr. Courtney Peterson is an associate professor in nutrition sciences at the University of Alabama at Birmingham and an internationally recognized researcher in the field of intermittent fasting and meal timing. Dr. Peterson conducted the first controlled feeding trial of intermittent fasting in humans and was the first to test early time-restricted eating, also known as early TRE, in humans. Her research has shown that early TRE introduces weight loss, alters hunger, improves glycemic control, lowers blood pressure, decreases oxidative stress, activates anti-aging pathways, increases autophagy, and improves metabolic flexibility, but does not affect energy expenditure, and that many of these effects are independent of weight loss. Currently, she is the principal investigator, or PI, MPI or site PI of 7 clinical trials on time-restricted eating, including a couple of the largest studies of intermittent fasting in humans. Dr. Peterson has clinical trials of TRE in patients with type 2 diabetes, prediabetes, cancer, and in healthy midlife adults to see if TRE can slow the aging process. She's also a consultant or collaborator on another 6 clinical trials on intermittent fasting. Her research has been featured in more than 100 media outlets, including NBC Nightly News, the Wall Street Journal, the New York Times, the Washington Post, Good Morning America, and the BBC. And today she is here with us. So thank you very much, Dr. Peterson, and I turn it over to you.
Thank you so much for having me. I'm going to load my slides. Well, fantastic. Thank you so much for inviting me to this amazing symposium. So I'm going to talk today and answer the question, do intermittent fasting and time-restricted eating promote well-being? Now, well-being is a broad term. Oftentimes we think about social and emotional well-being or health, but I would say it also just sort of encompasses the whole spectrum of health outcomes. So I'm going to review the data on a wide spectrum of health outcomes. And I have no disclosures. So intermittent fasting is sort of defined as any sort of meal timing intervention or approach that alternates periods of eating and extended fasting. And for my talk today, I'm going to use a 14-hour definition, meaning you have to be fasting for at least 14 hours at a time. And this is based on a recent international consensus committee that is now defining intermittent fasting as fasting for at least 14 hours, at least in humans. I also like this definition of 14 hours or longer for 2 reasons. So one, if you look at data from NHANES, which is one of the largest nutritional assessments of Americans, the median American currently eats over about a 12-hour period every day. So I'm reluctant to say that fasting for 12 hours is generally should be considered as fasting. And then second, if you look at substrate oxidation as a function of the number of hours someone's been fasting, we really don't see a big upregulation of fatty acid oxidation and gluconeogenesis until more like 14 to 20 hours of fasting. So I think 14 hours is a really good definition for practical purposes. So there are many types of intermittent fasting that have been studied in humans, and they're really quite diverse. So to give you a flavor of them, here are among some of the most popular forms of intermittent fasting studied in humans. Now, to make sense of all these different types of intermittent fasting, I like to kind of broadly group them into 3 different categories. The first type of category I call periodic fasting. This is not a widely recognized term in the literature, but bear with me. So this sort of category of approaches are approaches where you fast for 24 hours or longer, some number of times per week or per month. And examples of this would be fasting 1 day a week, which is common in many religious traditions, or alternate day fasting, ADF. This is where you fast, have a complete 24-hour water-only fast every other day. The sort of second category of approaches I would call intermittent energy restriction. So these are approaches that involve modified fasting. So instead of a complete 24-hour water-only fast, you're eating a very low-calorie diet. And we know eating a very low-calorie diet recapitulates many of the benefits of fasting. And examples of this would be alternate day modified fasting, where every other day you approximately eat the equivalent of 1 meal a day. the 5-2 diet, where you pick 2 days a week, either consecutive or non-consecutive, to eat a very low calorie diet, again, about the equivalent of 1 meal a day, or the fasting mimicking diet, which Valter has just walked us through. And the third sort of category of approaches are your daily intermittent fasting approaches, which are now known as time-restricted eating in humans or time-restricted feeding in animals. And in these types of approaches, you're compressing your sort of daily eating window into a smaller number of hours each day, and you're picking a consistent time to do that each day. So you can do that by either eating breakfast later in the day and or dinner earlier in the day. So you're kind of extending your sort of nighttime or overnight fast. So let's kind of go through some of the evidence that fasting or intermittent fasting can improve well-being. So let's start first by looking at fasting 1 day a week, which is common in many religious traditions. So one of the best studies we have on this is the Wonderful trial, which was done by Ben Horn. And this was a 26-week trial in 103 adults where they were either they either fasted 1 day a week. It was not a randomized study, but they either fasted 1 day a week or they were non-fasting. And they were followed for roughly half a year. Now, they found actually really good adherence in this study, so about 95% of adherence. And body weight wasn't quite statistically different between group, but you can see there was a statistical trend here for a sort of greater, roughly 3-pound difference in decrease in body weight. However, they did find improvements in insulin resistance as measured by HOMA-IR in the intermittent fasting group. And they also found improvements in the metabolic syndrome score here, as you can see as well. Unfortunately, they did not find any statistically significant improvements in brain-derived neurotropic factor or in scores indicating cognition. The second type of intermittent fasting that's been reasonably well studied in the literature is alternate day fasting, or ADF. So in this sort of approach, you alternate 24-hour water-only fasting with ad libitum eating. And back when the first studies on this were done, we really didn't know if intermittent fasting induced weight loss in the long term, because there's some possibility that individuals could compensate and then overeat, or at least eat enough to kind of bring them back into energy balance. So one of the first studies was done by my postdoctoral mentor, Eric Ravison, at Pennington Biomedical Research Center. And this was about a 3-week study in 16 people. And interestingly, they did find that alternate day fasting induced weight loss in sort of this zigzag pattern here. So it was sort of sustainable over the long term. People didn't eat enough on the ad libitum days to offset the weight loss. But interestingly, in the same study, they found that hunger levels essentially went through the roof. People were hangry. And if you read between the lines, they conclude that alternate day fasting is likely not sustainable. So we actually don't see very much research anymore on this type of intermittent fasting. Nonetheless, there are a couple interesting studies that I want to show you. So this is sort of the best quality evidence we have on alternate day fasting. So this, I'm going to show you results from a study published in Cell Metabolism, as well as 2 other studies. So in one of these studies, they took 60 adults without obesity, and they randomized them for 4 weeks to alternate day fasting or a control eating schedule. And they did indeed find that alternate day fasting reduced body weight. It also reduced blood pressure. It reduced heart rate. It also reduced the Framingham risk score. And there was some evidence as well that it might even improve thyroid hormones and increase ketone levels. In a second study published by the same authors, they did a sort of longer-term cohort study in 90 adults without obesity, and this was a 6-month-long study, so it's sort of more of like kind of a case control study. And they also found that alternate day fasting in the long term does reduce body weight, and it improves very similar CVD outcomes as their randomized controlled trial. But in addition, they found that in the long term, alternate day fasting lowered cholesterol levels and sort of other markers, including some certain like immune related markers. But interestingly, they found that alternate day fasting was a third study found that alternate day fasting, this is the Catenacci et al. Study here, found that alternate day fasting was not any better than CR suggesting that the most of the effects are actually from losing weight. The third type of intermittent fasting that's been appreciably studied in the literature is alternate day modified fasting, or ADMF. And really, Dr. Cristoverdi, who's at the University of Illinois at Chicago, is really the one who put this type of intermittent fasting on the map. And she did a lot of pilot studies in the early day on this type of intermittent fasting. And the way that she typically implements these studies is that she will have participants alternate fast days where they will typically eat about 25% of their daily caloric needs with feast days, which are ad libitum, and typically participants will eat more than they usually need to maintain their weight, but it's not enough to offset the calorie deficit on the fast days. One of the first RCTs that she did that was sort of larger in scale was this 12-week RCT in 32 adults without obesity. And she found that alternate day modified fasting does indeed improve body weight, as you can see here. And it does reduce fat mass and fat-free mass. And if you look at the ratio of the two, it's not selectively or negatively affecting fat-free mass, which you can think of roughly as skeletal muscle mass plus organ mass. In that same study, she also found that alternate day modified fasting reduced lipid levels and improved the inflammatory marker as C-reactive protein. And a little bit more recently, we also have evidence that other aspects that ADMF may improve other aspects of health. So in an 8-week-long RCT in 32 adults with metabolic associated fatty liver disease, the alternate day fasting did improve ALT levels. It reduced liver steatosis, and it also reduced measures of liver stiffness. And then even more recent, and around the same time, she also did a really elegant study comparing alternate day modified fasting versus habitual eating patterns and also versus CR. So this was a 3-arm randomized controlled trial in 100 adults with obesity. And they followed their assigned group for 1 year. So the first group basically was told to maintain their body weight, not change their eating habits. And those are these open circles here. The second group was alternate day modified fasting, these triangles here. And every other day they ate approximately 25% of their daily caloric needs. And then the dietary restriction group here, they were counseled to cut their calories by the same amount overall, but they were just asked to do that on a daily basis rather than on an every other day basis. And interestingly, what you can see here is they found absolutely no differences in body weight between the CR or calorie restriction groups and the alternate day modified fasting groups, although both groups clearly did much better than the control group. And they also found absolutely no differences in any cardiometabolic risk factors between alternate day modified fasting and CR. So there's no difference in inflammatory markers, visceral fat, blood pressure, glucose, insulin, and so forth. So then you could sort of conclude that alternate day modified fasting is equivalent to calorie restriction or calorie counting, no better or no worse. However, unfortunately, in this study, the dropout rate for the alternate day modified fasting group was a bit higher than for kind of standard calorie counting. So it did sort of suggest that alternate day modified fasting is not quite as feasible as classic calorie counting. There is a little bit of good news though. So alternate day modified fasting still improves health overall relative to baseline or not cutting calories. In addition, there's some other studies finding that alternate day modified fasting improves other health outcomes. So for instance, Cristoverdi's team also did this very nice post hoc analysis in the same group of 100 adults. And they did indeed find in individuals who are insulin resistant at baseline that they experienced greater improvements in insulin and HOMA-IR when they were assigned to the alternate day modified fasting group. And then another 8-week-long randomized controlled trial in 56 women with overweight found that alternate day modified fasting did improve some aspects of sleep quality. So it does improve sort of self-reported subjective sleep quality, and it also reduces daytime dysfunction. So which is sort of the issue of sort of sleep interfering with daytime functioning. So things like falling asleep while driving or while you should be awake during the day. The next type of intermittent fasting that I want to mention is the 5-2 diet. So this is an approach that involves eating a very low calorie diet twice a week. So to me, this is basically a less extreme version of alternate day modified fasting. Instead of doing this every other day, you just pick 2 days a week to have as your modified fasting days. The interesting thing is these days can be consecutive or non-consecutive, so it's up to patient preference. And this type of approach has actually over 25 clinical trials on it, so it's a really large number. I think when I first started giving these talks, we only had 4 studies on it, so it's just exploded in popularity. And it's actually most popular in Europe. You don't hear many people following it in the US, but it's extremely popular there based on a BBC documentary that was done many years ago. And one of the first studies that put this type of approach on the map was a study done by Michelle Harvey and Anthony Howell back in 2013. And they did a 3-month-long study in 115 overweight women with a family history of breast cancer. And they compared they compared the 5-2 diet versus continuous energy restriction, which is essentially just means the same thing as CR. And what they found is the 5-2 diet was actually better at reducing body fat levels, insulin levels, and insulin resistance than CR. And this sort of dovetails with some of those results I showed you earlier from Krista Verdi's lab. And so this was among some of the first evidence we have that intermittent fasting actually may be better than calorie restriction. There may actually be some benefits independent of how much you eat. Today, we now have 5 one-year-long randomized controlled trials with about 50 to 110 patients per group. So this is fantastic. We're starting to get to the place where we're having higher quality clinical trials. And the very interesting thing, if you look at these studies, is they all tend to report, well, actually, sorry, let me take that back. About half of them tend to report that the 5-2 diet induces greater weight loss around the 6 to 12 week mark. And here's an example from one of those studies where they compared a control group versus sort of classic calorie restriction versus intermittent fasting in the form of the 5-2 diet. And you can really see at the 12-month mark that the intermittent fasting group has done the best. However, once you get to the 1-year mark, all of the studies conclude there's no difference between the 5-2 diet and classic calorie counting. And the reason for this is the studies have all traced it back to waning adherence. People just tend not to be able to stick with the 5-2 diet long-term. And in fact, their adherence at the 1-year mark is about half of what it is at the 3-month mark. Now, the good news is the 5-2 diet in all these studies does improve cardiometabolic risk factors relative to either a control eating schedule or to baseline. So absolutely no question there that improves health. But relative to calorie counting, it doesn't seem to improve health any better or worse, with the sole exception that at least one study did report that the 5-2 diet improved postprandial C-peptide levels better than calorie restriction, so meaning that it improved insulin secretion. And then also that it reduces postprandial triglyceride levels, which are an independent risk factor for cardiovascular disease, better than calorie restriction. And now we're even getting evidence that the 5-2 diet may be helpful in the case of type 2 diabetes. So we now have 2 really quite large sort of 12 to 16 week long randomized controlled trials with sample sizes of over 100 per group, testing whether the 5-2 diet can improve glycemic control. And here's the results here, sort of on the right-hand side of the slide from the first study and the second study. Now, in the first study, they compared the 5-2 diet, shown in orange here, versus an exercise group versus a control group in gray. And they found that the 5-2 diet was superior, at least at the 12-week mark, in improving hemoglobin A1C. And then it also increased the fraction of patients who were able to achieve remission from type 2 diabetes, and you can see that here. And in another study, very nice study published in JAMA Network Open, another team of investigators compared the 5-2 diet combined with meal replacements. So it was a very clever study design. They helped participants adhere to the 5-2 diet by giving them meal replacements on that day, which I think is a brilliant way to combine the two. And they compared that with prescribing the patients either metformin or an SGLT2 inhibitor. And again, they found that that intermittent fasting actually even outperformed pharmacotherapy in this case, and similarly also increased the number of patients with better controlled blood sugar levels. And now we also are starting to get evidence that the 5-2 diet may improve liver health. So we now have a handful of RCTs, all between 8 to 12 weeks long with, okay, sample sizes, but they all universally find that the 5-2 diet may reduce metabolic associated fatty liver disease, as well as steatotic liver disease. And the types of changes that typically report are reductions in overall liver fat. And you can see here the 5-2 diet is performing just as well as a low carb, high fat diet. And there's also evidence that it reduces steatosis, fibrosis, improves indication, sort of liver markers, and then also lipid levels, and reduces inflammation. And also we're starting to see people, really, I've said, even in the last year or two, I've seen a huge increase in intermittent fasting to sort of treat cognitive disorders and improve brain health, which I think is very exciting. So this work, I believe, came out of Mark Matson's lab at the NIA, although I think he might be at Hopkins now, but they compared a 5-2 diet versus prescription for basically healthy living in 40 older adults who had insulin resistance. And through some really clever experiments, they were able to show that the 5-2 diet actually reduced neuronal insulin resistance, and they measured this through exosomes. It also improved the brain age score. It improved recall, and it improved other measures of cognitive function, including executive function. So very, very cool study. The next type of approach, which I will briefly discuss, which Valter did obviously a fantastic job on, so I'm just going to delve on a couple aspects of the fasting mimicking diet that he has not discussed, is the fasting mimicking diet. I sort of think of the fasting mimicking diet as a form of intermittent energy restriction, but as Valter discussed, it's actually a little bit beyond that, because you also have this prescription that it's a plant-based diet and that it's low in protein. But in any case, one of the first larger studies that Valter's group did was a study in 100 healthy adults, comparing the 3 cycles of the fasting mimicking diet versus basically a control group, which, didn't change their habits. And what they found in this study, as is no surprise, is the fasting mimicking diet reduced body weight and BMI. It reduced body fat. It also reduced visceral fat, hepatic fat fraction. In the main analysis, there were no differences in glucose levels, but in a subgroup analysis among individuals with prediabetes, it did decrease glucose levels. It also decreases IGF-1 levels, as Valter has said, and it improves blood pressure. And then further subgroup analyses have also found positive results for lipids and inflammatory markers. And another really cool thing that I'm, happy to present on is the fasting mimicking diet also appears to reduce biological age. So in this same study by Valter's team, he found the fasting mimicking diet reduced biological age by about 2.5 years. You can see that kind of by the shift in this curve to the left. And it also has improves the lymphoid myeloid ratio as well, which is sort of a measure of age. And then there've been some really cool studies looking at other aspects of health, sort of things that researchers don't study as often, but which are also super important to a lot of people. So there's also evidencing that fasting mimicking diet improves psychological health and even skin quality. So in one study, which is a 3-month-long RCT in 20 patients, they compared psychotherapy with time-restricted eating versus psychotherapy alone. And they found that the addition of time-restricted eating improved psychological health more than psychotherapy alone, which is fantastic. And it also improves self-esteem. Some of this may be driven by the weight loss involved. And these folks, I will say, they were followed up for a total of 6 months. So that could be a component there. And the other study I wanted to highlight, they actually looked at the effects on skin. So this was a 3-month-long RCT in 45 healthy adults. And they found that the fasting mimicking diet improved facial skin, and specifically it improved skin hydration and skin texture as measured by skin roughness. And they also reported that participants also reported that it improved their happiness and their confidence. And then lastly, on the fasting mimicking diet, there's also evidence that it may be really helpful for either inducing diabetes remission or reducing the need for medication. So we now have 2 one-year-long RCTs in adults with type 2 diabetes. And both of those studies reported that the fasting mimicking diet reduced hemoglobin A1C, which is a measure of glycemic or blood sugar control. And one of those studies even found remission in up to 47% of patients enrolled, which is fantastic. And then it also reduces medication use as measured by the medication effect score. So you can see here over time, patients are needing fewer medications, which is really exciting. And there was sort of a cost analysis of the savings from using a fasting mimicking diet, and it cut medication costs by about 77%. So that's fantastic. And then the last type of intermittent fasting, which I believe now is the most popular form of intermittent fasting, is time-restricted eating, or TRE. And interestingly, studies report that now between 10 and 12% of all Americans are currently practicing intermittent fasting. So that's very exciting. In time-restricted eating, sort of what that involves is eating within a consistent 10-hour or less daily period, so fasting for at least 14 hours a day. The most common form of time-restricted eating is the 16-8 diet, so 16 hours of fasting and then an 8-hour eating window. And what's sort of different about time-restricted eating relative to these other intermittent fasting approaches is that it involves counting time, not calories. So there's not a specific calorie prescription that you're trying to meet. So it's not like you're eating 0 calories a day or you're trying to target 800 calories a day. You're just counting time. And so what that means is the neat thing about time-restricted eating is you can practice it both with and without cutting calories. So you don't even have to do this for weight loss if you don't want to. Now, one of the first studies to really look at time-restricted eating was a study out of Krista Verdi's lab. This was a 12-week long study with historical controls. She tested an 8-hour window starting at 10 a.m. and ending at 6 p.m. in 23 adults with obesity. And what she found is that time-restricted eating did decrease body weight. The effect was about 2.5% in 12 weeks, which is less than what she typically saw with other forms of intermittent fasting. But one of the nice things about this is participants achieved this sort of weight loss without even trying, meaning they spontaneously lost weight. They didn't have to deliberately try to say, okay, I'm only going to eat 1 meal a day. They just naturally ate less and lost weight. And then the other thing is that adherence is really quite high for these types of intermittent fasting. So when they measured adherence, it tended to vary between about 5 and 6 days a week, but the average was about 5.5 days a week, or about 80% adherence. And we now have adherence measured in over 3 dozen studies, and it does seem that the average adherence is about 5.5 days a week, and that's been maintained for even up to a year. So we're really seeing good adherence and feasibility results with these types of intermittent fasting. We now have more than 125, probably even 150 studies now of TRE in humans. Now, most of the evidence we have is not very strong, meaning that the studies are either small, they're short in duration, many of them lack a control group. Though we are starting to see more and more studies breaking that 100 sort of person mark these days, which is exciting. Nonetheless, if you pull the results from all these studies, the last probably half a dozen meta-analyses have all consistently concluded that time-restricted eating reduces body weight. So here's data from one of those where they pulled the results of 17 RCTs, and they found that it induced about 2% weight loss relative to the control group. My lab also did a study looking at whether time-restricted eating could improve weight loss. And one of the neat things we did in that study is we actually wanted to use the weight loss data to kind of model and figure out what's the equivalent energy deficit that it induces, what's the calorie deficit it induces, because those of us who do diet research, you can give people food records and often they'll underreport what they're actually eating. But in any case, what we did in this study is we took 90 patients with obesity, and for 14 weeks, we randomized them to either practice time-restricted eating by eating early in the day, or to follow a control schedule where they ate over 12 hours a day or longer. And in addition, participants will all counsel to lose weight. So we were interested in the question like, if you're trying to lose weight, can time-restricted eating help you lose more weight? And here's what we found. We found that time-restricted eating did induce more weight loss. And you can see sort of the effects widening there. And this was statistically significant. And when we went back and modeled the data, we found the deficit induced was about 214 calories when we looked at all participants, so meaning even the folks who did not adhere to time-restricted eating. And then when we looked at the subset of people who really did adhere to time-restricted eating, which we defined as 5 days a week, every week, the effect was about 350 calories a day. So this is about a moderate or medium-sized calorie deficit. It's the equivalent to losing about 0.5 pound a week. So, nothing to sneeze at there. And then more recently, I will say there was a really nice study published in Nature Medicine. It was a 3-month-long RCT in about 200 adults with obesity. And in that particular study, they did food records and they similarly found that energy intake was reduced by about 250 calories a day. So it's equivalent to about losing a really nice rough rule of thumb is it's equivalent to losing about 1% of your body weight per month. And indeed, in this study, they found time-restricted eating, regardless of the time you practiced it, whether you ate early in the day, late in the day, or you self-selected your window, help people lose about 3% of their body weight in 3 months. So again, that 1% per month rule is very helpful. So we're seeing this replicated again and again, which is fantastic. Now, in the early days of time-restricted eating research, I was very interested in the question, why does intermittent fasting help people lose weight? Is it because it helps them burn more calories, or is it because they helps them actually eat less? Does it suppress appetite? So we did a 4-day-long randomized controlled trial in adults with overweight, where at the end of the 4 days, we stuck them in a metabolic chamber for 24 hours and we measured how many calories they burned. Unfortunately, we found that intermittent fasting does not help people burn more calories. There's no magic calorie burning effect. But on the flip side, we did find that it suppressed appetite. And we looked at both subjective and objective measures of appetite. And for instance, we found that time-restricted eating actually reduced ghrelin levels, which is a hunger hormone in the morning. So we did find evidence that it was affecting hunger hormones. One of the other really interesting things we found by putting people in that metabolic chamber is that time-restricted eating sort of alters these daily periods of fat burning. So we did find an increase in total fat burning, but we also find this general pattern that if you put someone on a time-restricted eating diet, during the daytime, they're not burning more or less fat than someone who's on a normal diet. But starting at night, for us, it was around, 8 p.m., 9 p.m., we see actually a big increase in fat burning in the middle of the night. And actually, this is the control group. So lower values here means more fat burning, but we see that the body is really shifting to this fat burning mode. And that's really important because conceptually, we think a lot of the benefits of intermittent fasting are from the fact that we're just kind of extending or dipping deeper into a sort of fat burning mode. We're really altering sort of the underlying metabolism and physiology that sort of people go through on these daily cycles. Now, a related question concerns body composition. How does TRE affect body composition? It's great that it induces weight loss. Could it potentially negatively affect muscle mass? And we find absolutely no evidence it does. We find it does slightly decrease total body water. So there are a couple studies reporting, lower effects on lean mass, but it seems to be solely due to reducing total body water, which has been measured separately. And moreover, in studies looking at things like muscle protein synthesis, you can see here there's absolutely no negative effects of TRE on muscle protein synthesis. And then there have been several studies, at least half a dozen, looking at the effects of time-restricted eating on either muscle mass accretion during resistance training or in strength gains. And there was absolutely no negative impact on the ability to build muscle and no negative impact on the ability to gain strength or muscle performance. So for instance, if you look at pre and post measures in those who are on time-restricted eating versus a control diet, you can see both are about equally gaining strength during a leg press and also during a bench press. So we really don't see any impairments there, which is great. We now have about 4 one-year-long RCTs of time-restricted eating, and all report really good adherence around about 6 days a week. So unlike other types of intermittent fasting, TRE does seem to be more feasible. In one of these studies, they actually compared TRE versus calorie restriction versus the control group. Again, this is from Krista Verdi's lab. And the interesting thing they found in these individuals as compared to the control group. TRE did reduce body weight, and it was about 4.5% in this study. I think it was about 4.5. And it also improved cardiometabolic endpoints. But relative to calorie restriction or calorie counting, it was no better or worse. So you might take that as bad news, but actually take that as good news, because most people do not like counting calories. And so time-restricted eating becomes an alternative way to lose weight and get the benefits of calorie restriction without actually having to count calories. And now there's also evidence that time-restricted eating can improve cardiometabolic health. We have about a half dozen studies now in patients with type 2 diabetes, and they pretty much universally all report improvements in glycemic control. The largest of these studies was done in China and 120 adults with type 2 diabetes. And in that particular study, they found that eating in a 10-hour window improved glycemic control as measured by hemoglobin A1C. It also reduced insulin levels, HOMA-IR levels, and reduced medication use. And then other studies have similarly reported improvements either in hemoglobin A1C levels or in other metrics such as time in range. So when people wear continuous glucose monitors, we can actually measure their glucose levels over the course of the day. And our goal there is to keep them in a tight range. We don't want those glucose levels to be too high. And what we see with time-restricted eating is it actually reduces a lot of those spikes in blood sugar levels. And in this particular study, they found improvements in 24-hour CGM levels in the absence of weight loss. So also very encouraging. There's also data that time-restricted eating may improve metabolic syndrome and prediabetes. So in one study, one of the first to look at the effects in patients with metabolic syndrome, it was a 12-week single-arm study in 19 adults with metabolic syndrome, and they found that TRE reduced body weight, BMI, waist circumference, visceral fat, and blood pressure, as well as some lipid levels. And then in another study, one of the first in patients with prediabetes, they took 101 overweight adults with prediabetes, and they randomized them to TRE or a control group for 3 weeks, and they found that 8-hour time-restricted eating improved body weight, fasting glucose, triglycerides, and HDL cholesterol. My lab a few years ago was interested in the question of whether TRE can improve cardiometabolic health independent of weight loss. Because one of the big questions in the field is, it's nice that it's improving health, but how much of this effect is just simply due to losing weight? Is TRE just a fancy way to lose weight? And so to test this out, we did a 5-week-long crossover randomized controlled feeding trial in men with prediabetes. And we tested time-restricted eating using a 6-hour window, which is pretty extreme, but this was sort of more of a proof of principle study versus a 12-hour eating schedule. And essentially what we found is that eating in a 6-hour period early in the day was much better for reducing insulin resistance. It lowered blood pressure by about 10 points, which is pretty dramatic. Although I will say most of our participants also had hypertension. It also improved beta cell responsiveness, which is sort of the ability of the beta cell to produce insulin, and it reduced markers of oxidative stress, similar to what Valter Longo has mentioned for the fasting mimicking diet, though we found no effects on lipid levels. And now today we have a total of 8 studies that report TRE has benefits independent of calorie restriction. In fact, I think I can think of only one study that found no benefit, but just to know our results have been replicated and extended by other groups. For instance, one particular group, Jones et al., they found that 2 weeks of early time-restricted eating, similar to what we studied, improves insulin sensitivity as measured by glucose uptake into skeletal muscle, as well as branched-chain amino acid uptake into skeletal muscle. And here's some of their results for insulin sensitivity. And then we've also done other studies in our lab. We were one of the first to look, I think we were the first to look at 24-hour glucose levels as measured by CGM. And we found that only 4 days of practicing time-restricted eating by eating early in the day improved 24-hour glucose levels, as shown here by this sort of thick black line. And interestingly, we sort of originally thought, well, most of the effects will probably be due to the time of day of eating or the longer fasting duration. But it actually looks like perhaps most of the effects are actually due, if you look at the second lunch meal of the day, it looks like a lot of the effects are due to eating meals in succession before glucose levels return to baseline, and that you get this sort of second meal effect whereby you know, you already have excess insulin in circulation. So if you start your next meal before insulin levels have returned to baseline, that actually can reduce the spike or subsequent glycemic excursions. There's also evidence that TRE can improve some, but not all aspects of cardiometabolic health. And where we see the most promising data is for blood pressure and some related aspects of health. So for instance, gosh, I think we now have 24 studies of time-restricted eating. And meta-analyses now consistently conclude that time-restricted eating does reduce blood pressure. And this includes studies even in patients with hypertension. And then there was a really nice study that was published about 1.5 years ago in patients who recently experienced myocardial infarction. And they actually found that time-restricted eating actually improves left ventricular function, as shown here. And then a very nice study that I was a collaborator on in older adults, that was a crossover study, found that time-restricted eating actually improves the 6-minute walk test and actually reduces heart rate while exercising, which a couple other studies have found as well. So it seems to somehow also improve physical function in some ways we don't understand that are independent of exercise, at least in older adults. So I think that's a very cool area worth watching. In terms of effects on lipids, we really don't see a big improvement in lipid levels unless TRE is also accompanied by greater weight loss. So if you look at all the studies out there, about 75% of them report absolutely no effects on lipid levels, or at least fasting lipid levels. But there is some evidence that TRE may affect other aspects of lipid metabolism. So one study in 24-hour shift working firefighters did find that TRE positively affected VLDL particle size. And then another study, which was actually done in primates found that TRE actually reduces HDL, actually improves HDL size and content and improves cholesterol efflux. And that was true in both sort of younger adult monkeys here, which is these open bars, as well as in geriatric monkeys. So very nice result there. And then lastly, I also want to talk about what we more traditionally think about as measures of well-being. So there's evidence that time-restricted eating can also improve things like mood, quality of life, happiness, and self-esteem. So we were one of the first to take a look at this. We did a 14-week-long RCT in 90 adults with obesity, and we found that time-restricted eating actually improved vigor activity as measured by the PALM short form. So you can think of this as sort of self-reported energy levels. And it also reduced fatigue and then sort of feelings of depression and dejection. And in a per protocol analysis, we even found later that it reduced feelings of anger and hostility, which we were quite surprised about. And it also reduces sort of total mood disturbances. Now, it's not clinically reversing folks who have depression and making them depression free, but it is still shifting people more in the right direction. And there have been some other studies also finding some encouraging results in this space. So for instance, there was a 3-month-long RCT in 30 healthy adults where they had patients follow an 8-hour time-restricted eating schedule, and they found that time-restricted eating improved quality of life. It also reduced fatigue, both mental fatigue and physical fatigue, and it improved, reduced pain, mental and physical function, pain, mental and physical, pain, physical health, and mental health, as well as a couple other constructs, including health perception. That said, there are some quite notable studies of time-restricted eating that have reported either null results. So for instance, I can think of a famous study published in JAMA Internal Medicine, where they had participants practice TRE by skipping breakfast. And those participants ate later in the day and stayed up later at night, and they found no benefit from time-restricted eating. And there are even some studies that have tested TRE quite late in the day. So for instance, by having individuals skip breakfast and lunch. And some of those studies have actually reported negative effects. So one of those studies, again, done by Mark Matson's group a long time ago, they randomized participants to either eat 1 meal a day starting at 5 p.m. in the evening. So this would be kind of like late in the day, very late in the day, or to eat 3 meals a day. And they actually found that eating that 1 meal a day late in the day actually worsened glucose levels, insulin levels, blood pressure, and cholesterol levels. So this suggests that the time of day that you practice time-restricted eating may affect at least some aspects of health. And this makes sense because we now know the body has an internal biological clock known as the circadian system, which produces 24-hour rhythms in our physiology, metabolism, and behavior. So for instance, your best coordination and greatest muscular strength and fastest reaction time are in the afternoon. Your body starts to produce the hormone melatonin in the evening, which helps you sleep. And then your sort of greatest alertness and testosterone production. And in most people, their best glycemic control is in the morning. And in fact, we've known for over the last more than 50 years that if you give someone an oral glucose tolerance test in the morning, say at 9 a.m. versus the afternoon and the evening, despite the fact that you're giving individuals the same load of glucose, blood sugar levels spike significantly higher in the afternoon and the evening, suggesting that oftentimes when we're eating dinner, individuals who otherwise have normal blood sugar, many of them are equivalent to being pre-diabetic. So borderline pre-diabetic at dinner. So just to say, like, that dinner meal may actually be really important in terms of what people are doing. Or said another way, we should probably try to avoid eating large meals or unhealthy meals quite late in the day. And in fact, we are starting to see more and more studies testing time-restricted eating by eating early in the day versus late in the day. And I'll say at the moment, the data is kind of mixed. I will say a majority of those studies find no major differences in weight loss, but they are finding differences in other aspects of cardiometabolic health. So some of those studies, like the one I'm showing here, do report greater improvements in glycemic endpoints, such as fasting insulin and HOMA-IR, with early time-restricted eating. And they're also finding greater improvements in blood pressure with the early time-restricted eating as opposed to the late time-restricted eating. So we'll continue to watch this space. I think it'll be interesting. So to sum up, and give you some high-level take-home points, I would say, first of all, really important lesson, I think not all forms of intermittent fasting are feasible. As we've seen, some of these are really hard to stick with in the long term. I think if you were to ask my opinion, I think the three most promising are probably the 5-2 diet, the fasting mimicking diet, and time-restricted eating. Second, I think the data in the literature clearly suggests that intermittent fasting induces weight loss and improves cardiometabolic health, at least relative to sort of habitual eating patterns. Intermittent fasting may also combat several age-related diseases, in particular type 2 diabetes, fatty liver disease, hypertension, and potentially even cognitive decline. Now, when you compare intermittent fasting to calorie restriction, the data suggests by and large that intermittent fasting isn't much better, with some exceptions. I think we are seeing some evidence that time-restricted eating, at least with an early eating window, may be better than CR, or certainly better than any sort of calorie-matched approach. But by and large, intermittent fasting seems that most of its benefits do seem to be from reducing calorie intake. And then lastly, there's evidence that intermittent fasting can improve energy levels and fatigue and improve mood, quality of life, mental health, happiness, and confidence/self-esteem. And with that, I'd like to thank all the folks who were involved in generating some of the data from my lab included in this presentation, and then thank also our grant support and our sponsors, including the NIH and Department of Defense. All right, thank you so much.
Thank you very much. That was a wonderful talk, very informative and engaging. And I think it's also going to set up our afternoon talk. And I know that because I'm the next talk.
Welcome back, everybody. I'm Dr. Andrew Heyman, the director of the Fellowship for Integrative Medicine here at George Washington University. And I have the privilege and the honor to introduce Dr. Frame. She has accomplished so much in her career. I have to decide which bio, and I think this one it best encapsulates. So Dr. Frame is Chief Wellness Officer of George Washington University Medicine and Director of the Resiliency and Wellbeing Center. She also serves as Executive Director of the Office of Integrative Medicine and Health and Associate Professor of Clinical Research and Leadership and Physician Assistant Studies. As co-founder of the Resiliency and Wellbeing Center, she's advanced resilience, whole person care, and well-being initiatives across GW and beyond. With extensive experience in biomedical research, Dr. Frame bridges nutrition and immunity through translational research. Her interests include the microbiota, gut-brain immune interface, malnutrition and obesity, vitamin D as an immune modulatory hormone, research ethics, science, communication, resilience, and whole health. She consults for and serves on global healthcare and biotech boards. In 2021, she was named among the top 100 healthcare leaders by the International Forum on Advancements in Healthcare. Dr. Frame graduated with distinction in biochemistry from Mary Baldwin College, now Mary Baldwin University. She earned her MHS in molecular microbiology and immunology and her PhD in international health, human nutrition from Johns Hopkins Bloomberg School of Public Health. While pursuing her PhD, she gained expertise in obesity and metabolic health through work at Johns Hopkins Center for Bariatric Surgery. You know, I read all of that and I think you probably listen to that and think, gosh, did I really do all that? And, you know, you've already had an amazing career. So I know everybody's excited to hear what you have to say.
Sorry about that. My Zoom meeting controls went away and then I couldn't figure out how to unmute myself. I am back and very glad to be here. Okay, so what we're going to talk about today is just kind of an extension of what we've been talking about, but I think maybe putting it in a slightly different context, which hopefully will be helpful for both clinician and individuals alike. My conflict of interest slide, nothing affects the contents of this talk. Okay, so we're going to talk a little bit what I think of kind of as the missing piece of the fasting discussion so far, and sort of why fasting works on a biological level in a bigger sense, as opposed to more the nuanced sense that we've been talking about so far. So I want to put this in context. Here's a nice little visual here, sort of the beautiful symphony that is the human body. And we have all sorts of inputs, like for instance, the light dark cycle that set our circadian clocks. And another one of these is food intake. And so that's what we're going to focus on here. Why is food intake important for our circadian rhythms? And really, fasting, while it does have caloric restriction, and we've talked about all the benefits of that, it's not just about that. It's also about synchronizing our biology. And this involves the gut microbiome, the liver, and metabolic processes, which we're going to dive into here. And my hypothesis is that the real power of fasting may actually be how it interacts with our biological clocks as opposed to the simple caloric restriction. Okay, so the gut microbiome, we're going to start here. It's one of my favorite topics, and it does have its own daily rhythm. But it doesn't see the sun, right? How does it know whether it's day or light? Well, I'm going to tell you that it's a fasting regulated ecosystem. So the gut microbiome has this daily rhythm, and as part of that rhythm, you actually see a shift in bacterial species depending on the time of day and the fasting state. And it's really that fasting that's going to shift that composition and thus the function of the gut microbiota. What are they doing at certain hours of the day? You're going to see certain bacterial species are actually going to peak at different times of the day. So they're not all changing in the same way. Some are going to go up, some are going to go down, and that's going to maybe reverse in other parts of the day. Some really thrive in that fasting state. And so for them, it's important to have some fasting period of time to kind of grow up and become strong. And when I talked about the microbiota, I talked about what are they doing, right? They're making metabolites. And metabolites like short chain fatty acids and bile acids and polyamines have all been shown to fluctuate, one according to who is there, but because who is there is shifting over the day with a daily rhythm as well. So there's actually a daily rhythm of these metabolites in addition to the different bugs that are actually there in the microbiome. Oh, well, this is new. Didn't know I did this. Okay. So we've got different ways of thinking about fasting. And what we've really talked about was, we talked about how we're either eating or not eating. But for this talk, we're going to focus more about like, when are you eating? Are you eating during your active period? And if you're eating during your active period as a human, that means during the daytime. we would call that aligned eating. So that's aligned to your natural eating cycle. We would typically eat during the day. However, if you're a mouse, your active period is at night. And so thus aligned eating would be at night. Now, if you eat when you're not typically active, so in the middle of the night for a human or in the middle of the day for a mouse, that's misaligned eating. And misaligned eating can just be sort of late night meals, or it could even be an erratic schedule. If you don't have a regular rhythm to when you eat, that can actually disrupt your body's clock because it's not really sure when to expect food. And that changes how many things are functioning in the body, not the least of which being bile acid release from the liver, which we will talk about. And because you have this misaligned eating, you actually see a disruption in the balance of the gut microbiome. What you see is a promotion of inflammation and metabolic disease. And what does that actually look like? So if we look over here, we could see in the fed state, there's a potential for imbalance, right? You could be overfeeding, or you're really skewing things towards the microbiota that don't flourish in this fed state. So remember I said some people actually do better in the fasting state, some of these microbes. And so you could potentially be having an imbalance if you are only in the fed state. And what we typically see is carb and fat digesting bacteria are really going to grow up during the fed state. And none of this is bad. It just has to be put in balance with the fasting state. So what happens during the fasting state? You actually see more of these short chain fatty acid producing species, which if you listen to my previous Sung Symposium lecture on microbiome, you'll know is one of the key elements of linking the gut microbes and fiber to health outcomes. And so short chain fatty acids are something that is produced by our gut microbiome when we eat fiber. And it also is not only important for them, obviously they're metabolizing it, but these short chain fatty acids are actually fuel for the colonocytes in our gut. So the cells of our gut cannot function without these short chain fatty acids. And so here's a reason where the fasting state could be really important. It's a time of repair, right? You have the colonocytes, they're uptaking these short chain fatty acids, they have fuel, now they can repair themselves, they become strong and prevent leaky gut. We'll talk a little bit more about that, but that's one of the really key things that why it has to be a balance is because you have to both feed and bring nutrients in, but you also have to repair. And like I said, some of the elements of the microbiota, certain taxa or phyla, are increased during the fasting state. So it is really a balance here. Akkermansia is one you may be familiar with. So one of the stars is Akkermansia municipala. That increases during fasting. So if you are never really in a true fasting state, you will have very low Akkermansia, and that has been linked to negative health outcomes. So I will add that is very non-specific at the phyla level. We're talking about very broad groups of bacteria. Okay, so we're going to walk through a couple of studies. This is the first one. And the title is Diet Diurnally Regulates Small Intestinal Microbiome Epithelial Immune Homeostasis and Enteritis. So this is an animal model. We're going to look at some animal model studies. And this is actually their visual abstract here. So each one of these panels we're going to walk through individually. So we know that certain cells are what we call light entrained, right? So light is telling them it's active or inactive time period. And that's really important for things like the brain, the suprachiasmatic nucleus in the brain. But like I said, the gut's not seeing that. So what is causing the rhythmicity in the gut? Well, what we find in this animal model, and I will show you it in other models as well, is it's nutrient intake. So if we have low nutrient intake, we have sort of the equivalent of the dark cycle for humans. And then if we have high nutrient intake, then it goes to this active state, right? And so we have this up and down with fed and fasted daytime and nighttime. And that is really what's signaling to a lot of the elements that don't have this light section as to whether it is day or night. And so really those gut rhythms are depending on meal timing. And so many of you have probably experienced if you travel, particularly if you travel around the world, you get jet lag and it's terrible. And those type of symptoms are what we are talking about when we are talking about misalignment, right? You are misaligned when you have jet lag. And one of the ways to prevent jet lag, well, there's many ways, but one of the ways is to try and time your meals more to the area you are trying to get to, right? So wherever you are trying to move your cycle to time your meals on that time, and that will help with jet lag. Same thing with light, getting light exposure on what you are trying to move your time cycle to. Okay, so that's number 2. And because of that, we see this change in the microbes that are present in the microbiome. So the composition of the microbiome also shifts with day and night. And we'll talk a little bit more about that in detail. The next one is that because you're having this shift in, let's say it's active or inactive period, what else is happening is that it's actually triggering a change in the microbiome immune reaction. So the way that the gut microbiome is perceived by our immune system. And so you see a different activation of the immune system. And so during well, this is actually reversed because this is mice, but during the inactive period, during the time of rest, the immune response actually gets higher. So the immune response over here is actually getting higher, and then it gets lower during the day. And we'll talk a little bit about that in detail. But essentially, it's sort of this balance, right? You don't want to have too many microbes, but you don't want to have too few. And so having this oscillation allows a maintenance of balance. And what you also see is a change in a reaction in terms of what cytokines are actually produced. So IL-10, for instance, is what we typically would refer to as an anti-inflammatory cytokine, and it has sort of the opposite rhythm to immune sensing. So if immune sensing is peaking during the inactive period, IL-10 is going down, and vice versa. So here we actually want to have the immune system reacting, so we're turning down this anti-inflammatory IL-10 to actually spark an immune response. And what we find is that misaligned meals are actually disrupting this. They're disrupting this, and this cycle is lost. And when you take an animal model that has had this disruption through misaligned meal timing, it actually increases their susceptibility to enteritis. So they're more prone to this pathogenistic outcome simply by changing the misaligned meal timing. Everything else is held constant. So light's held constant. All of the things that we maybe would typically think of when we think about circadian rhythm are held constant, but food is changed. But the best thing is when you bring back them into aligned meal timing. So you shift away from the misaligned and you go back to an aligned meal timing. You actually can reverse that. It promotes gut resilience. So it's something that is reversible. And again, any of us who've ever been jet lagged, you understand it is reversible. It is very, very uncomfortable when you're experiencing it. But the problem is if we have prolonged exposure to that. And so we'll talk a little bit about that later. But first, let's talk about what else they found in this study. So they looked at gene expression. Here's just a little bit of a 101 for those of you to make sure you know what we're talking about. So we have DNA, right? DNA is basically the template from which we make things. And so we transcribe the template. Here is the message. And then that message is translated into proteins, which actually are doing things. They're the enzymes, there's the structural proteins. And so this is what we're talking about when we're talking about gene expression, is this process. And in this study, looking at the mice, and they actually looked at the gene expression in the small intestine, so locally in the small intestine, they found that over 1,600 genes showed changes in their expression patterns that were cyclical due to circadian rhythms from meal timing. So a diurnal variation that was indicative of either whether it was aligned or misaligned meal timing. It was following that pattern. And because of that, if you are controlling the feeding time, you're essentially controlling the peak expression of these genes, all 1,600 of them. And here are some example genes just to get a feeling. So Reg3 and DEF-A, which are antimicrobial peptide related genes. So, okay, what are we talking about here? Antimicrobial peptides are part of the immune response. So you could see why it would make sense for this to be on a fed-fasted timing, because we want a stronger immune response while we're fasting, because we're doing that repair and recovery. And we want a lower immune response when we're feeding, because it's more important for us to be processing the food and getting the nutrients from the food. Another one is claudins and other tight junction proteins. So I kind of alluded to this, right? Leaky gut. The reason that a gut can be leaky is because those tight junctions between the colonocytes, they're weakened for one reason or another. Maybe the colonocytes don't have enough fuel and they can't repair themselves, and those junctions become leaky. And they're important for that barrier function and protection from pathogens like enteritis. And so what happens is when you have feeding, it actually suppresses the tight junction, not feeding. When you have misaligned feeding, it suppresses the tight junction proteins. So if you're feeding, well, actually both is technically true. That's why I'm struggling here. So let's talk about just feeding generally first. So when you're eating food, you want to get the food from the inside of the gut to the outside of the gut, right? You want to be able to absorb it. And so there is some downregulation of the tight junction proteins to facilitate that. That being said, you also want to then turn that off, right? As soon as you've absorbed as much of the foods that you need. And so that's important. But then misaligned feeding is sort of causing more of a general downregulation of tight junction proteins, what we would traditionally think of as a leaky gut. So it's getting turned on generally. So you always have this downregulation of a tight junction protein instead of it going up and down with meals in a more health promoting way. Okay, so now we're going to look at this mouse in terms of its feeding. So I already told you the inactive phase is the daytime, the active phase is the nighttime. I know it's kind of confusing because it's the opposite of us. So we're going to just talk about aligned versus misaligned. So if we have these aligned feedings, what we saw was oscillations in the microbial composition. So we saw things like lactobacillus or Bacterata changing with the inactive versus active phase, depending on whether they were being fed or not. And misaligned feeding basically undid a lot of that, right? So you typically would have a microbial rhythm, but when you have misaligned feeding, that's kind of flattened. It's sort of changing the difference between the fed and fasted state because they're not happening at the right time. What else did we see? We saw a drop in diversity, which if you know anything about my talks, you know that diversity is the sort of most agreed upon marker of gut microbiome health, which we can't even define what a healthy gut microbiome looks like. But this seems to be the one marker that sort of generally is health promoting. And so we don't want diversity to go down. That's a negative thing. And then metabolites also lost their rhythmicity, including short chain fatty acids. So again, there's not as much of a difference between the fed and the fasted state. It's all kind of blunted. And the same thing then with the immune activity and the ability of the immune system to actually control that microbial growth in a balanced way is also blunted. So it's still there. It's not completely inactivated, but it's just not as strong as it needs to be. So essentially, these feeding fasting cycles, they're gonna train the microbiome, right? They're gonna say to the microbes, this is what you should be doing now, this is what you should be doing later. And it's this cycle that they kind of get into. And this is why it's important to have rhythmicity of eating, right? There's an on period, there's an off period, and having it generally be regular is really important for them to kind of get into that cycle, or resetting the cycle if they are off that cycle. And the microbes themselves are actually important for training the immune system. So this is just an example here of how the microbiome can interact with the immune system. And this is what we call immune education. So the majority of your immune system at any one period of time is residing in your gut. And that is because the gut for the gut or for the immune system, it's basically like grad school. They are going to learn higher level information. They are learning whether this is a state that is generally inflammatory and they should be ready to fight, or if everything is kind of normal and they're just out there looking and surveilling and making sure that everything is okay. So that's what's going on here. So you can see if you have an alteration in these microbes and the response that they're the message they're giving to the immune system, that it could be detrimental, sort of largely speaking, not just in the gut, because the immune system is then going to travel over across the whole body and use the information that it learned in grad school throughout the body. This, however, is still being elucidated. So this is kind of early research. And there are certain cells that have shown to be particularly involved here. And I'm not going to get into details on this other than this one particular cell type, it secretes antimicrobial peptides. So that's something we're going to hit on time and time again during this talk. You will see that comes up many times. And there's actually a rhythmic release of the granules that hold the antimicrobial peptides in these cells, and it's triggered by feeding. So the only way to alter that timing is by having a fed state and a fasted state and having it be regular and rhythmic. Similarly, goblet cells, they're mucus producers. So this green area here, this is mucus. It's a really important part of how the gut microbiome functions. It keeps enough distance away from the immune system that they're not constantly on alert and being like, oh my gosh, they're invaders. And so it's really important to have enough mucus. And we found that in these mice, when they were misaligned, the mucus producers suffered. They did not have that same rhythmicity as you would expect to see. And so there's alterations then in that mechanism as well. So here's sort of a general idea of what this model looks like. So they used a DSS-induced colitis. So this is basically they're giving a chemical to induce colitis in this mouse model. And sort of generally speaking, what happens there is you see damage to the colonocytes, right? You might have a little bit of leakiness, things are getting through. The mucosal layer is gone. See, over here's a nice healthy mucosal layer. There's no mucosal layer here. So basically, they've kind of undone a lot of the control that is typically there between the gut microbiome and the immune system, and things are really off the rails. And what happens is they develop colitis. And when you have this model with misaligned feeding, you're basically adding some gasoline on that fire. So what do you see? You see more severe enteritis. You see a higher epithelial damage, so more of this damage with misaligned feeding. And then you see immune dysregulation, which sort of makes sense, right? Like they're here at university and they're trying to learn, but basically they're in the middle of a war and they aren't really learning what they need to learn. And so their response is often inappropriate. But the good news is they were actually able to rescue them with time-restricted eating. So if you get them back onto an aligned feeding regimen with a restricted window, that actually restored that protective gene expression, those 1,600 genes, and the microbiota rhythmicity. So it came back, right? The increase in certain bugs and the decrease in the others, and then the flow of short-chain fatty acids and other metabolites is able to be restored when they implemented time-restricted, or you would call it time-restricted feeding in mice. It's time-restricted eating in humans. But it's the same concept, right? So this could be a mechanism for overcoming guts that might have a pro-inflammatory status. This may be beneficial, but obviously we're talking about mice here, so this is still early phases. All right, the next study, this is another mouse model. So again, we're in the early phases, but I think this one's gonna blow your mind and it's gonna be really worth it. And here we're looking at sort of the mechanism behind feeding-dependent regulation of the intestinal barrier function. Okay, so this study replicated some of the findings of the previous one. They found that the gut microbiome composition did have a diurnal variation, this rhythmicity, that was driven by fed and fasted state. So again, it's in mice, but I will tell you that later we're going to talk about that it's not just in mice, but it's just much easier to study these things in mice sometimes. And over here is actually the data for that in case you are interested. So they have a number of different types. Basically, fasted is here and fed is here. And you can see really there's a pretty clear difference between the fed state and the fasted state. There's even more so over here. And this is with small numbers. These are early studies, but I think the mechanism is so strong behind these, it would be surprising if we aren't able to continue to replicate this. And so this is the part I'm really excited about. This is actually microscopy of the gut. So these little buds here, these are the villi, the little fingers of the gut. Well, go back to one of those pictures, right? So they've got these little fingers here, these projections. And what that does is it gives more surface area for the gut to absorb nutrition. So it's a really important indicator of the gut, by the way. And so here we've got these little fingers, and what's going on with these little fingers? Well, they have a little bit of these bacterial threads. These are basically chains of bacteria. There's a little bit of them here, and there's lots of them down here, and they're much longer. And the only difference between these two groups is this group is fasted, and they have the stubby chains, and this group is fed, and they have the long chains of microbes. And so this is really showing us what we're talking about, right? Like the immune system during the fasted state turns on and it knocks down the microbes in the gut microbiome, kind of controlling them, saying, okay, that's enough growth for one day. And then when you go back to the fed state, they can grow back up again. And it's this cycle of up, down, that's really important for helping to control, because you don't want too much of a good thing, right? Like if these guys just kept growing and kept growing, then you'd have microbial overgrowth, which is a known problem. So we don't really want to be promoting that indefinitely. So it's important to have a fasted state. And how does this work? Well, it works through the immune system, not surprising. And IL-22 is one of the big ones. When we feed, we get out of the fasted state. IL-22 production is actually reduced. And when it's reduced, that causes a decrease in gene expression. So this one should look somewhat familiar. Hey, it's an antimicrobial peptide. So we're having the peptides that are sort of like antibiotics for within the body, right? So they help reduce the count of these bacteria, and it's decreased because we're feeding and we're less concerned about knocking down the bacteria. Right now, we want to do more of the absorbing nutrients. And with that, you see this change in morphology, right? So you see it going from stubby to long, and altered composition as well, which we talked about earlier. Now, what's really cool is they've got these two conditions here. So this condition is the one we just talked about. The only thing that's different here is fasted versus fed. Over here, they delivered an anti-IL-22. So they're sort of blocking the IL-22 production without the use of feeding. So it should be somewhat in the middle, right? So it's sort of like having, here we have a fasted state, but you're blocking IL-22, so it's not as much of the fasted signaling is getting through. And so what you see is a little bit more growth here, right? There's a little more growth than there is here. And down here, it's a little hard to tell because there's already so much growth here, but there is a little bit more growth here than there is here as well. So the IL-22 really disrupts the ability of the immune system to, or the anti-IL-22 disrupts the ability of the immune system to control microbial growth. Okay, so what happens when we feed? There are actual mechanisms behind this. So enteric neurons, these are basically neurons, right? Brain cells that are in the gut. And it's part of the gut-brain axis, the communication that the brain has to the gut and the gut has to the brain. And when we feed, these neurons actually activate a specific pathway. And what that pathway does is it inhibits that IL-22 production. So this is the mechanism behind IL-22 working. And IL-22 then reduces the antimicrobial defenses. It also increases greater nutrient absorption, which I've alluded to, but we haven't specifically mentioned. And in this particular study, they specifically showed that lipids were poorly absorbed when you had IL-22 production, or when you had decreased IL-22 production. It also increases the enteric pathogen risk, right? So that's why we're talking about enteritis. There's a higher risk for developing gut inflammation and pathogenic infections. But it's important to balance this. This is all about balance, right? So we've got this neuron activation, right? That's because we just fed. We also have a change in the immune response. So IL-22 is turned down. The ILC3 cells, these are immune cells, are actually changed the way they work. The microbial control is loosened, and that increases the pathogen susceptibility. And so, but we also have this trade-off here with nutrient absorption. So we want to loosen the microbial control so we can enhance our nutrient intake. And so it's actually a trade-off between defense and absorption. As with many things in the body, we sort of need to do one or the other. I like to tell people that you can either digest or rest. Right, you need to eat your food, digest it, and then go to sleep and get a good night's sleep so you can switch to the repair. And this actually is very similar, right? You can't do both. You can't be feeding and absorbing nutrients and be repairing and knocking back things and having the immune system actually respond. And so it's about this balance. You don't really want to be always fasted, but you also don't want to be always fed. And having this sort of ebb and flow is really natural to the way our immune system functions and the way we control our gut microbiome. Okay, so let's just talk about in brief, what does this mean for TRE? So in health, what we find is that it increases microbial diversity. We see beneficial metabolites are produced. It seems to be beneficial for the immune system. It sort of works with the immune system. And then it's also beneficial for metabolism, which we're going to talk about a little bit more right after this. But first, we're going to go to the liver. Liver, also a fasting regulated system. So this is going to be set up very similarly. When we have a fasting state with the liver, what happens? It's detox enzymes are activated. It moves to a fat breakdown priority, so you're burning fat, and it's releasing glucose or causing ketogenesis. So obviously in the feeding state, it's going to be very different, right? So those detox enzymes are inhibited. It's not time to repair. It's not time to clean things. And we're going to be creating fat. It's time to store, right? We just fed, let's store fat. And the same thing for glucose. We're going to store that for later. So what happens in this process? So if we ingest carbohydrates, we have an insulin response that involves the pancreas, which by the way is also a fed-fasted circadian rhythm. So we'll talk about that. Then you have the uptake. The liver is actually utilized, so the liver is activated. And here's really part of the reason that the liver is so important here, because the liver becomes active in response to food, right? It doesn't come active in response to sunshine. So when you are feeding your body, your liver says, it's time to work. And so this is really one of the key here. And then you can, use your muscles and your fat tissue and everything else around it. But it's this key here that the liver needs to be utilized and it needs to be awake. And similarly, you might be having ketosis. And so for those of you who don't really know what ketosis is, basically it's a way to take fat and turn it into a form of fuel that can be used by the brain, because the brain really is more of a glucose user. And the only way it can use fat is this ketone synthesis, which occurs when we are fasting. And so typically when you are fasting overnight, you will enter ketosis and your brain will be running off these ketones, which allow the brain to function in a very different way. And it's switching to that repair mechanism, which we'll talk about. However, we have this issue here with overnutrition. So a lot of people in our world don't ever fast, right? We always have food around us. It's very difficult to fast. Many people even get up in the middle of the night and eat. So now you're not even having that overnight fast. And it can really disrupt the whole system. This continuous fuel delivery, it can actually alter the mitochondria, the way the reactive oxygen species are produced, our proteins, and even lead to cell injury. And we'll talk a little bit more about that. So what are the metabolic benefits here in fed versus fasted states? So if we maintain the fed state, what's the pros? Well, we're going to have rapid energy, stable blood glucose. The tissues can grow. We're in that strength building stage. We won't have hypoglycemia. We should have metabolic flexibility in that we've kind of got whatever we need sitting around to create whatever components we need. What are the cons? The potential for overconsumption and possible weight gain, right? If we're constantly eating, it's a little harder to control. This is why we're talking about TRE as being a way to restrict calories. There is potentially a risk for insulin resistance, obviously, depending on what type of food you're eating. There is a potential for nutrient imbalance. Are you having too many of a certain nutrient? And then you're going to lose some of these fasting benefits. Okay, so this is one of the big reasons why we don't want to maintain a fed state, is there is a reason to be fasting. What goes on in the flip side in the fasted state? So you do have energy, and that's linked to our stores as well as ketogenesis. Brain function support. So the brain is getting the ketones that it actually really enjoys taking a break. It's like practicing metabolic flexibility, and it's allowing itself to repair. It's also important for muscle protein. So this can be quite preservative. We don't use the muscle protein as fuel as long as we have enough ketones. Regulates inflammation, reduces oxidative stress. But there's cons to that too, right? So we might have energy dips. We could have hypoglycemia. There's definitely a risk for nutrient deficiencies if you are fasting too much. There could be an adaptation period where maybe you're not feeling so fantastic at first. It's also not suitable for everyone, which we're going to talk about in the next talk. And there is a risk of muscle loss if it's not managed appropriately, but really we're talking about extended fasts there and less so time-restricted eating. So I think I've alluded to this. It's about balance. It's the yin and the yang, the fed and the fasted state. You're either having protein synthesis, ketone production, a prolonged fasting, or chronic insulin elevation. None of these are particularly good, right? We don't always be in protein synthesis even. Like we need to move from one to another and have breaks, and having that balance is really what is key. So what happens to the liver when we're fasting specifically? So we start fasting and it immediately begins to adapt because the liver is very adaptive. It will switch to ketones if needed. It will also begin to cleanse itself if we have longer fasting periods, that autophagy that we've been talking about. And then it switches to the fat oxidation, right? So it's actually breaking down fat for energy. It will also be detoxing itself. So that's really important. And that mitochondria undergo that alteration in the way that the liver is utilizing energy production will happen sort of at the later phases of fast initiation. So if you start fasting and then you maybe get down here to, okay, we start to dip into some of our ketones, but then you pop back up, right? That's what we're talking about. You don't get down to the autophagy and any of the other benefits. There is a period of time you need to be fasting in order to receive these benefits. And Dr. Peterson talked very nicely about that. So what is going on with the liver? So it's got circadian rhythms. It has core clock components. These are actually regulators of that cycle. And there's an interaction with the liver and the brain, not surprisingly. They're talking to each other, saying, what's up? What time is it? Is it eating time or is it sleeping time? And that actually changes the feeding behavior, and that can be disrupted if we are not on an aligned feeding schedule. So a little introduction here to my favorite friend, the vagus nerve. I love this photo because you can see how this nerve really, it goes everywhere. It's called the great wanderer. That's what vagus means, because it goes all over the body. It's really important for sensing. It goes into the gut, it goes into the liver, the pancreas, all over the place. And in this study by Woody et al., they showed that in mice, the vagus nerve is actually part of this control of the liver food intake and that brain connection. So if you've got a mouse and it's just eating normally, the liver's on a normal circadian rhythm, it signals to the vagus nerve and the hypothalamus, and everything's good. We're all in alignment, right? The brain clock is the same as the liver clock. But if you have a dysregulated or a misaligned eating behavior, what happens is the liver clock is on a different time than the brain clock. And that dysregulation makes a lot of the negative effects happen, is that misalignment between the brain and the liver. It's the liver's trying to do one thing, the brain's trying to do another. Like the brain's trying to rest and repair while the liver's trying to make new proteins or all that sort of stuff. And what is really interesting is you can actually, what we call rescuing this phenotype by ablating or dissecting the vagus nerve. So you're cutting the connection between the liver and the brain, and it restores some of the negative effects in mice at the very least. So again, the vagus nerve and that connection with the brain is going to be part of the effect here. And it's the disconnect between the two that can be quite problematic. Okay, so let's talk a little bit of the gut-liver axis. Why are they connected? So we've got the irregular eating. This disrupts the gut microbiota. That also changes the bile acid signaling, right? So the liver is producing bile acids in a way that is not typical or aligned with our cycles. And this increases our risk for metabolic disorders like insulin resistance, fatty liver disease. That is what we're calling a disrupted gut-liver axis. Okay, so how do you reset this? What is going on? Well, fasting is what we'd call as a fancy term is a Zeitgeber, which basically just means it's a cue that resets the circadian clocks. And there are many different kinds of Zeitgebers, right? There are ones that are we call first order, which is like the sun and temperature, and second order ones, which are like food intake, right? So they are different and they interact differently with the body. Light is for the brain. Food intake is for what we call peripheral clocks, the liver, the gut, pancreas. These are all peripheral clocks. They're all driven purely by food intake. And so fasting is a Zeitgeber. It's a way to reset that chronobiological clock. And thus, it's a tool. It's something we can use to realign our clocks with day-night cycles. If we are eating within aligned windows, that should, in theory, produce all of these benefits, right? We eat in an aligned window that synchronizes our clocks, that enhances metabolic efficiency, and may also have some of these benefits of improving glucose regulation, supporting immune function, and optimizing health. And in theory, it's as simple as not eating when it's nighttime. It is a little bit more complicated than that, and there is more nuance, and it depends on the person, but that's a good rule of thumb, is if the sun is out, it is okay to eat. So if you're hungry, it's always okay to eat. Okay, so how do we weigh the pros and the cons of time-restricted eating? So pros, it improves metabolic efficiency, glucose regulation, it can enhance immune function, it's getting us on a good circadian balance. It's probably one of the biggest pros. And then there's no calorie restriction needed, right? We don't have to count calories. We've already heard that today. The cons, potential social limitations, right? Are you able to go out for dinner with your friends, or maybe you can convince them to have dinner a little earlier? There may be an adaptation period where you're kind of slowly moving towards a smaller and smaller window. If you're not typically used to doing this, that would be my recommendation, is don't just change yourself abruptly because you're going to shock the system. It's possible you might be hungry, and maybe the window is too long for you if you are having that experience, at least at this moment. That limited eating window can be psychologically challenging, which I'm hoping the last talk will talk on a little bit. And it really isn't suitable for everyone. So I wouldn't use this as a blanket tool. Okay, what else is going on? The immune system and brain health. So I alluded a lot to the immune system. We're going to fly through this because of that reason. But essentially, aligned feeding promotes a healthy immune system. And disrupted is disrupting the immune system. So you really need to keep that in mind as we're moving through some of these details. Okay, so when you have aligned feeding, what happens? We have an enhanced immune response and effective surveillance. What does that mean? What is another way of thinking of that? It's promoting homeostasis. And during homeostasis, we have immune surveillance and a robust response if triggered. So it's kind of just there and waiting and ready to go. But it's not turned on. It's not chronic inflammation, right? So ideally, this is what we want the immune system to be in. So aligned feeding promotes really an ideal state of the immune system. And it also supports immune cell timing. Well, what does that mean? So there's actually circadian-driven migration of certain immune cells. So neutrophils, macrophages, and T cells are triggered to move throughout the body by this circadian rhythm, by this fed-fasted pattern. And because we are aligned and our immune system is in this homeostatic way, that reduces chronic inflammation, right? It's not just willy-nilly turned on as it would be in chronic inflammation. And because of that, can aid recovery, right? You're in a rest and repair phase instead of a inflammatory response phase. Not surprising, misaligned is basically the opposite, right? So we're disrupting that circadian rhythm, we're impairing the immune response, increasing infection risk. It promotes inflammation-related diseases, and there is some indication that tumor formation risk may increase by misaligned eating. All right, what about the brain? So the brain, we already alluded to. Autophagy is really important in general. It's really important in the brain too, right? So we're having cellular cleanup, reducing neuroinflammation. And if you know anything about neurodegenerative diseases, these are both really important in there. So I'm not saying that fasting can necessarily treat neurodegenerative diseases, but it may be something that is at least able to slow or prevent those diseases. Metabolic flexibility, right? We're able to shift back and forth. The brain, it has what it needs. The body has practiced doing it, and that switching is really important. Inflammation, we have an improved insulin sensitivity because we have a reduced inflammation, and an inflammatory response that is controlled. It's utilized in the correct way. And what we see now is neuronal protection. This is actually protective. So we see an increased integrity in the neurons and a resilience against stress. And this actually includes BDNF. So many of you who listen to my mental health talks will know that brain-derived neurotropic factor, BDNF, is linked to many different mood disorders and mental health generally. We can promote it with physical activity, we can promote it with a healthy diet, and we can promote it with time-restricted eating. So this is another mechanism by which you can understand how fasting can promote enhancing brain health and resilience. Generally, what are the pros and cons? right? We're talking about reducing inflammation, metabolic fastability, autophagy. Here it is, may prevent neurodegenerative disorders. Big old may on that one, right? That this is early research, but it's one of those things that there's pretty low risk, so it may be worth trying. We know about the insulin sensitivity, which, by the way, may be one of the mechanisms that's helping support this neurodegenerative diseases. So there's certainly something here to investigate. The cons, I already said it's not for everybody. There's the potential for nutrient deficiencies, particularly in older individuals where, for instance, protein intake needs to be higher and is often insufficient. So now if you are having a more limited window to take all that protein in, it may be difficult to get all those nutrients in, but perhaps having a less restrictive window could overcome that. There is the risk of overeating post-fat. Some people have kind of a rebound eating after the fast, so that's something to be aware of individually, but also as a clinician. Possible initial discomfort. That's definitely true, but I will say there's also often some long-term benefits, and it does require strict adherence. Okay, so here are your practical takeaways. So the big principles from this is that you want sun-aligned eating, right? The sun's out, it's good, you're good to eat. A consistent eating schedule, that regularness of eating allows your body to be on a consistent schedule and a rhythm that it will be very happy with. Early caloric intake, I didn't even need to make this point because Dr. Peterson made it for me, which is fantastic. Avoiding late night meals, it's kind of the flip side of that, right? You don't want to be eating right before bed because that's when you're supposed to be resting and repairing, not digesting. And then having a good overnight fast, right? You're mostly going to be sleeping during this anyway if you're getting your 7 to 9 hours. And so hopefully this is just something that you can kind of do for an extra hour or two than you are already doing. In conclusion, fasting works not just because of caloric restriction. It's because it synchronizes our biological rhythms. And it's really important that we eat in an aligned way for many different things, including our metabolic and immune health. And aligning our feeding windows with that natural circadian rhythm appears to be a powerful tool for health and longevity. So there is much more research needed, though. There's some novel approaches here I'll suggest that I'm not going to get into because I'm short on time. But I do want to just, we aren't there yet. We don't have all the answers, but I just wanted you to see some of these mechanisms so you can understand maybe why TRE could be a good tool. And of course, I have to put a plug in for the Integrative Medicine podcast, which those of you who don't know about it is freely available anywhere you listen. And with that, I will close and stop sharing. And please keep putting your questions in the Q&A feature. And I am going to go ahead and introduce our last speaking group, a dynamic duo. Dr. Samantha Han, PhD, MPH, RD, and Dr. Jesse Bach, PhD. First, I'll talk about Dr. Jesse Bach. He's an associate professor of biochemistry and nutrition, the Department of Foundational Sciences at Central Michigan University College of Medicine. He obtained his BS at the University of Hawaii Manoa in food science and human nutrition, and his PhD at the University of California, Davis in nutritional biology, which I have to say is an excellent school for nutrition. So kudos to you. He completed his postdoctoral training at St. Jude's Children's Research Hospital. Professionally, he is a member of the Michigan Medical Nutrition Education Consortium and the treasurer for the Association of Biochemistry Educators. He has expertise in cell signaling, particularly kinase and phosphatase signaling in metabolism. Dr. Samantha Han is an associate professor of nutrition and epidemiology at Central Michigan University College of Medicine and a registered dietitian. She obtained her BS in nutritional sciences and human biology from Michigan State University, her master's of public health in nutrition and dietetics, and PhD in nutrition epidemiology and nutrition intervention from the University of Michigan School of Public Health. Following her graduate work, she completed an NIMH T32 postdoctoral fellowship in eating disorders epidemiology at the University of Minnesota. Her work in eating disorder prevention, and particularly her work aiming to understand how clinical and public health practices impact eating disorder risk, has been recognized by international organizations, including being inducted into the Eating Disorder Research Society. So with that, I welcome you, and I really look forward to your talk.
Thank you. Dr. Han, can you go ahead and make that full screen for us? While she's trying to get that into presenter mode, I'll go ahead and get started. So I'm very happy to be here today with you guys. Quite surprised to see the big number of people that are online. And I think it's really important topic. So today we have me and Dr. Han are going to be talking about eating disorder risk with relation to intermittent fasting. I do not do eating disorder research. My research is much more cancer focused. But I kind of want to tell my story of how I became aware of this other side of the intermittent fasting or time restricted eating. So go next slide, Dr. Khan. So overall outline of our talk today is I'm going to talk about my personal change of perspective, and then we're going to hand it over to Dr. Han, and who's really the expert on eating disorder field in general, where she's going to introduce eating disorders, talk about the overlap of intermittent fasting and disordered eating, if there is any, what data we have right now, and then how can we consider disordered eating when recommending or testing for intermittent fasting. So my personal introduction, or really the reintroduction. So here's my, I had a very kind introduction from Leo, where my bachelor's and my doctorate are both heavy nutrition focused. And so you'd think that someone with my education would be kind of well aware of the disordered eating and eating disorders within, changes in eating habits. But that's not the case. And I've been teaching human metabolism to students for a long time. I have a lot of students interested in eating intermittent fasting. I've been personally doing intermittent fasting myself for over 10 years, so probably 14 years now. And so I have a huge interest in this. And yet I had a major blind spot in relation to some of the risks of intermittent fasting or time restricted feeding or time restricted eating with patients. So one of my students approached me, wanting to publish a review paper on intermittent fasting. So we did that, just addressing it towards clinicians, one of the most classic areas of intermittent fasting that we heard about earlier today is about its role in diabetes prevention. And there's a lot of strong evidence suggesting that it does play a role, and it makes sense scientifically, it makes sense signaling wise and metabolically. And so we published that, and then right after we published that, go next slide. Dr. Han joined our university as a brand new assistant professor out of her postdoc. And we got talking because we're friends. And I very quickly realized I had a major blind spot. I did not consider that some of these patients, that these clinicians, that all my students are going to be talking and doing motivational interviewing, that kind of stuff, and talking to them about intermittent fasting and other changes in eating behavior, that I was not considering the risks of disordered eating. And so I very quickly with Dr. Han, we did an amendment to our publication. There was a commentary, making sure that we do discuss the risks of disordered eating in these populations of people. So with that, I will hand it over to Dr. Han. So hopefully your story will be similar to my story, and that we get to have a little bit of the realization that not everything, even though signaling wise is a lot of roses on that side, is scientifically time restricted eating is very beneficial in a lot of respects, but there is a little bit of a consideration that we need to make on our personal lives, and especially if we're going to be advocating for this type of eating behavior to others that may not share the same risk factors as other people. Dr. Han?
Thanks, Jesse. So I am going to be kind of talking about IF from an eating disorder perspective. So I want to give you my perspective in terms of my training and everything like that. I was trained in an eating disorder lab. I kind of came into nutrition like everybody else. I did an undergrad in human biology and nutrition, and then I did my PhD in nutrition and everything like that. But I was trained in an eating disorder lab. And so I think that really makes a difference in terms of how I saw nutrition. So I am a registered dietitian, but I do think it's important to acknowledge that even in my early aughts of training, I was trained in an eating disorder lab. So all of my my training in terms of how do we promote healthful eating came from this eating disorder perspective. My dissertation, my postdoc, all of that is really looking at how do we promote healthy eating while simultaneously preventing harm. But excuse me, my daughter is sick and she gave it to me, so I apologize if I am hard to understand. Because not all of us were trained in eating disorder labs, I just want to make sure that we're kind of all on the same page in terms of eating disorders 101. So, eating disorders are psychological disorders characterized by extreme beliefs, attitudes, and disorders surrounding food, weight, size, and/or shape. They have very strict diagnoses in terms of DSM, disease and statistic manual diagnoses. But these, excuse me, eating disorders have extremely high morbidity and mortality. So, folks who are struggling with an eating disorder are really struggling in all aspects of life. They are struggling in terms of like everyday aspects, what they're dealing with on a day-to-day is really hard. And so, I'm so sorry. What they're dealing with on a day-to-day in terms of morbidity is far beyond what we typically think of. And then in terms of mortality, eating disorders have a far higher mortality rate than people really think of. So eating disorders have the second highest mortality rate of any um, uh, mental health illness, second only to opioid use disorder. So opioid use disorder has the highest mortality rate, but then eating disorders, specifically anorexia nervosa, has the second highest mortality rate of any eating or any mental health illness. And when we think about what causes eating disorders, it's actually very complex. We can't kind of signal one thing as causing eating disorders. It's a range of biological, there's genetic components, psychological, there are certain traits that make folks more or less likely to develop eating disorders, and then sociocultural. So things like engaging in social media, or believing that having a certain body is more or less ideal. Those things all impact somebody's likelihood of developing an eating disorder. But again, they are far more common than people typically think. So about 9% or 30 million people in the US population will struggle with a full syndrome eating disorder in their lifetime. But many, many, many more will have disordered eating. So what do I mean by disordered eating? Disordered eating is really this kind of encompassing term of engaging in eating disorder behaviors, irrespective if somebody meets criteria for a full syndrome eating disorder. So many people will engage in disordered eating, things like fasting, which is particularly relevant for today's conversation, purging, compulsive exercise, binge eating, disordered muscle building behaviors, but they might not meet these diagnostic criteria for an eating disorder. So as I mentioned, eating disorders have these full threshold diagnostic criteria of engaging in these specific behaviors a certain amount of times and a certain amount of period. For example, it's three binge episodes in 3 weeks, for example. So just because somebody engages in 2 binge episodes in those 2 weeks doesn't mean that they're struggling less. It just means that they aren't meeting these very strict criteria for an eating disorder. So when I talk about disordered eating and eating disorders today, I'm going to be kind of broadly referring to disordered eating. I'm going to be talking about folks who are engaging in these eating disorder behaviors, irrespective of if they meet these criterion for a full syndrome eating disorder. Because irrespective of if they meet these full syndrome eating disorder cases, they're still suffering. So everybody with disordered eating is struggling, but not necessarily everybody who has a full syndrome eating disorder meets criterion for, excuse me, not everybody with a full syndrome eating disorder, I said that backwards again. I'm so sorry. Not everybody with a full syndrome eating disorder, not everybody with an disordered eating meets criterion for a full syndrome eating disorder, but everybody with an eating disorder is engaging in disordered eating. Irrespective, people who are engaging in disordered eating are struggling. They are suffering both mentally and physically, because disordered eating is both mentally and physically cumbersome. And to give you an idea of some of the consequences of eating disorders and disordered eating, just so you can see how severe it is, I want to kind of lay out some of these criterion. So some are psychosocial in terms of shame or guilt. Everybody who engages in disordered eating has these shame or guilt cycles, and engages in withdrawal from friends or activities. So folks who are binging are trying to hide those types of activities from other people, and they have decreased educational attainment. So folks who have disordered eating are less likely to go on to engage in educational activities in terms of going on to college and even going on to a full-time employment. There is decreased cardiopulmonary activity, low pulse and blood pressure, and even on the opposite end, high blood pressure, abnormal electrolytes. So this is something that we have to worry about, particularly for folks who engage in binge eating episodes. So we have to worry about fasting, and then afterwards worrying about how those individuals are engaging I'm so sorry. How those individuals are engaging in binge eating episodes, and then afterwards refeeding episodes. So individuals who are particularly starved are more likely to have abnormal electrolytes afterwards. And then musculoskeletal decreased body fat and muscle mass is particularly common among individuals with anorexia nervosa, but low bone density is common among individuals with anorexia nervosa, but also not anorexia nervosa, because the calcium and vitamin D deficiencies that are particularly common after folks engage in binging and purging. Those folks are more likely to have low bone density, even if they do not have low body mass index, which is important to note. Marked changes in malnutrition and body fat. So even folks who are not engaging in or do not experience low body weight can still have major changes in body fat and malnutrition. So just because somebody is not low body mass index does not mean that they are not malnourished. And that's an important note that I think in particular folks who are in the nutrition field, we tend to think about folks who are malnourished as being low body mass index, but in particular in eating disorders, it's important to think about folks who are across the weight spectrum. And then skin, gastro, and other endocrine disorders. Gastro in particular is very important because folks who have eating disorders often kind of present in terms of the nutrition world in non-primary care places. They often present in gastroplaces in terms of having indigestion, slow GI presentation, etc. And that's a very important place to kind of flag if this is happening. Is it really an eating disorder, or is it these other types of things that are happening? The other thing I want to highlight is that in eating disorders, we often think about what we call the swag stereotype. So in eating disorders, we often think that skinny, white, affluent girls are who has eating disorders, but this couldn't be farther from the truth. Eating disorders affect everyone. And in fact, they actually affect people who are very displaced from that swag stereotype more often than not. So they affect all genders and sexes and all sexual orientations. And in fact, LGBTQ folks are more likely to develop an eating disorder than heterosexual cis folks. All races and ethnicities, and in fact, folks who are BIPOC are more likely to develop an eating disorder, particularly binge eating and compensatory behaviors than white folks. So white folks are more likely to develop, behaviors like skipping eating or skipping meals and those type of behaviors. But again, binge eating and compensatory behaviors are more likely among BIPOC folks. All financial backgrounds. So my interest is actually in understanding eating disorder risk in low SES populations and particularly rural populations. And what we found is that low SES populations are more likely to develop eating disorders. So again, counter to that swag stereotype of skinny affluent folks. Affluent individuals are far more likely to receive diagnoses and far more likely to receive treatment, but they are less likely to develop eating disorders, counter to that swag stereotype. And then all sizes and shapes, again, counter to that swag stereotype. Individuals who are larger bodied are far more likely to develop an eating disorder. In particular, binge eating and compensatory behaviors are far more common among individuals of larger bodies. And then all ages, which I'm actually going to talk about in a little bit. But I think it's really important to highlight here that there is a disproportionate burden on marginalized groups. And again, that swag stereotype could not be farther from the truth. But how common is disordered eating? In high schools, so using their youth risk behavior surveillance system, 15.2% had fasted, used diet pills, or vomited in the 30 days prior to survey. So a huge proportion of adolescents are engaging in disordered eating. And then when we look at this kind of transitionary period from high school to adolescence, we see another large increase. So first year college students, we saw that 15% had fasted for 24 hours or more. So not just the, intermittent fasting kind of period, but 24 hours or more. 58% had skipped meals, 12% had purged, meaning they vomited, used diet pills or laxatives. 52% had excessively or compulsively exercised, and 51% used supplements of some kind, specifically protein powders, steroids, or another muscle building kind of product. And when we look at the undergraduate and graduate population more broadly, 49% of women and 30% of men had binge eaten, and 31% of women and 29% of men had used a compensatory behavior. But I think it's important to recognize that disordered eating, yes, we definitely think of adolescence and young adulthood as this kind of peak period, because certainly it is when we see this period of the highest prevalence. But there's also this kind of bimodal characteristic. in midlife, where we see another peak of disordered eating and other types of unhealthy weight control behaviors. And the reason that we see this is, broadly, when we think about disordered eating, we are thinking about diet culture. We are thinking about the influences of why somebody might engage in this type of eating or weight-related behavior. And the big driver is this underlying idea that thin is good and fat is bad. In particular, this really is becoming kind of quote unquote true or resilient in these bimodal characteristics. So in adolescence and young adulthood, we have a lot of people who are engaging in like things like social media, and they have a lot of people telling them like, you should look a certain way, your body should look a certain way, and if you just try hard enough, you can look that way. And if you don't look that way, it's your fault. And you can use makeup or filters on social media, etc., to meet this beauty ideal. But the underlying idea is this thin and muscular ideal is good and fat is bad. And by and large, when we're seeing these bimodal occurrences, is actually when we're seeing deviations in our natural ability to meet these ideals. So in adolescence, particularly girls, they're gaining adipose tissue, right? They're going through puberty and they are getting more adipose tissue, and that is taking them away from this ideal of thin, of if I just achieve super, super thin, then I will, quote unquote, be attractive. But this also happens later in life, right? Folks who are going through menopause or later in life, they are having hormonal switches that are making them more likely to gain adipose tissue around their midsection, more likely to gain adipose tissue irrespective of where they gain it. And this is drawing them further and further away from this like ideal of a curvy ideal, a thin ideal, and more towards this like quote unquote fat is bad. And they think more and more that this fat is bad is because they are doing something wrong. But in reality, we all know as researchers that our ability to lose fat and maintain fat loss is so hindered on genetics, on really everything, right? So we are not able to maintain weight loss in the sense that society tells us we should be able to maintain weight loss. And so we internalize this inability to maintain weight loss as our own personal failings. And so we think we are bad because we can't maintain this weight loss. And in particularly later in life, when folks are in particularly focusing on intermittent fasting and trying to lose weight, they are not actually biologically capable of losing and maintaining this weight loss, and they internalize it as their own moral failure. So now I want to talk about kind of the overlap of disordered eating and intermittent fasting. And I know this is going to be kind of controversial, and I welcome any and all thoughts, but disordered eating is harmful eating and weight-related behaviors specifically to change weight and or shape. And that includes fasting. And then intermittent fasting, on the other hand, is skipping meals and fasting, this time-restricted feeding. So you can see that they do overlap in many senses. So when done for weight and shape purposes, intermittent fasting is by definition disordered eating. And I know, again, this is going to ruffle some feathers, but it is this definition of the motivation that folks are having. Why are they intermittent fasting? Is it for weight and shape? Is it for metabolic health? This is all very dependent. And we know that in other kind of fields like physical activity, the reason that somebody has engaging for, say, physical activity has an extreme effect on the outcomes associated with that behavior. So, for example, physical activity is associated with very different health behavior outcomes if somebody is engaging in it for weight and shape purposes versus kind of these day-to-day effects in terms of, I want to feel better. But it's also important to know that irrespective of if we considered intermittent fasting disordered eating, IF can also lead to other forms of disordered eating be above and beyond that fasting component. And particularly this restriction and binge cycle. So in this restriction, you have a low calorie or food limited groups. Again, this is going to be that restriction, folks engaging in that restriction specifically for intermittent fasting. But for some folks, that leads to feelings of feeling deprived or hungry and feeling an increased urge to eat. And that can actually lead to episodes of binge eating. So feeling out of control around food or unable to stop eating. In particular, a binge eating episode is characterized by two things. First, eating a large amount of food, an objectively a large amount of food, and then two, feeling a sense of loss of control during that eating episode. And in fact, we have seen in my own research that intermittent fasting has led to binge eating episodes. So this was a mixed method study where we followed folks in over time to see how weight-related self-monitoring, so things like using MyFitnessPal, Fitbit, impacted their disordered eating risk. And folks in this mixed method study, particularly the qualitative portion, so the interview portion, reported that when they were engaging in time-restricted feeding, and particularly intermittent fasting, that they felt so kind of restricted and like they were restricted not in terms of just the quantity but the quality of their eating, that they went on to engage in binge eating, whether or not they called it binge eating. And then beyond just our qualitative research, which I know some people have some qualms about in terms of, is there temporality? Did the binge eating actually, or did the time restricted feeding actually lead to the binge eating? We have another study by Dr. Ganson and folks in the Journal of Eating Behaviors, and they looked at a general population. And this is, again, a cross-sectional study, so we're not able to assess temporality, but they found that in the general population of emerging adults, about 50% of women and about 40% of men engaged in time-restricted feeding or intermittent fasting in the last year. And in the last 30 days, it was about a quarter of folks. So intermittent fasting is not just this clinical tool anymore. It's not just this very restrictive, we're getting so much instruction about intermittent fasting. It is now transcended into the general population, and folks are engaging in intermittent fasting with or without the tools that they need to do so safely, because 50% of women are engaging in this type of behavior. But it's also important to note that intermittent fasting in this general population was associated with high rates of eating disorder risk. So in the population of women specifically, we saw that folks who had engaged in intermittent fasting in the last 12 months were more likely to overeat have a loss of control eating, engage in any binge eating, vomiting, laxative use specifically for weight and shape control, have any compulsive exercise, fasting, or kind of a global EDQ score. That would signify that they were high in eating disorder risk. So in particular, I want to draw your attention to laxative use. So folks who engaged in intermittent fasting were 3 times more likely to engage in laxative use compared to those who had not engaged in laxative use. So irrespective of how somebody is engaging or whether or not it is causing an eating disorder, we are seeing very, 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 very high rates of disordered eating among those who are engaging in intermittent fasting. In fact, 3 times the rates. And this might be because of a population overlap for sure. We're not able to look at causality in these types of research studies. So IF is recommended for weight loss among those with larger bodies for sure. We've heard here today that IF is particularly good for weight loss, and people do end up losing weight if they are engaging in intermittent fasting. But we also have to think about kind of this overlap. And those with larger bodies also have higher eating disorder risk. Again, counter that swag stereotype. Folks with larger bodies have higher eating disorder risk, which means they are more likely to engage in disordered eating, and they are more likely to have things like binge eating episodes as a result of intermittent fasting. So kind of contradicting our own stereotypes and thinking about who has eating disorders and disordered eating. It's also important to note that dieting is a potent predictor of future eating disorders. So when we look at who goes on to develop eating disorders, dieting is the #1 predictor. Folks who go on, or folks who are dieting, are the most likely to go on to develop an eating disorder. So if we are recommending dieting in some sense, that is problematic. And we can think about the folks that we are recommending dieting in the form of IF are the most likely to go on to develop an eating disorder or disordered eating. So we need to kind of think about our follow-up with those folks. And because of popularity and reported benefits, IF is just not solely in the clinical and supervised spaces. My line of research is really looking at how do clinical, these clinically used tools impact the general population in terms of calorie counting and intermittent fasting. We recommend them at an individual level, but because we are so ecstatic about the potential consequences and the potential use of these things, they often transcend into our own individual recommendations. So it's not just the people that we are recommending use these things. It's also the general population that is hearing about how great these tools are, and then they are going on to use these things. So we need to think about, how does our messaging impact not only the individuals that we're recommending, but also the broader population that we may be recommending to. We also have to think about, among those with high levels of eating and weight-related concerns, those are probably who we are recommending IF to in terms of folks with diabetes, folks with higher body weights, folks with hypertension. Again, these are not just individuals who are left in their own in society. They live in this weight stigmatizing culture. And so they are being told that their body is the problem. They have their own feelings and thoughts about their weight, and they are likely being fed the messaging if not by their own selves, by other individuals, their doctor, their loved ones, that they need to lose weight. And so if we are thinking about intermittent fasting and who we're recommending that to, it's probably also the individuals who are told they need to lose weight and therefore are getting all of these other messages that could increase their eating disorder risk. But irrespective, I think this really boils down to whoever we are giving intermittent fasting advice to on a clinical level, on a population level, we need to be very, very careful on if and how we recommend IF in terms of we have to think about the potential consequences. For sure, there are benefits, metabolic benefits, as Jesse has taught me many, many times. But I also think about the flip side of the coin, right? In terms of who might this harm? And we can't just think about the benefits. We also have to think about the harms on an individual level, but also population level, right? And in particular, I can say in our medical students often hear about all these potential benefits of intermittent fasting and they think it's just all good, right? And I think that's kind of consequential of all of the messaging around intermittent fasting. It's all beneficial, but in reality, we have to think about this flip side of who might it not be helpful for. And really advocating for those folks, because I think oftentimes the folks that are not advocated for in terms of who it might be harmful for are who's not receiving advocacy in general. So I already talked about this, but consider your broad messages when communicating benefits of IF in terms of this is just like an AI generated news article in terms of communicating, oh, IF is great. I think IF can be great, but we also need to make sure that when we're communicating the benefits of IF, that we are also telling people it might not be beneficial for X, Y, and Z folks for this reason. We want to make sure that we're not just saying it's great for everyone, because it's not. If you're a clinician, I really advocate for universal screening and, in fact, rescreening of folks with disordered eating. So if you're advocating for intermittent fasting, we want to make sure that we're not advocating for intermittent fasting for folks who are struggling. And I included two screeners here, the ESP and the SCOFF. The ESP is 5 questions. SCOFF is also 5 questions. They both have very, very high sensitivity, not so much specificity, but that's okay. I would rather have high sensitivity than specificity. But there are yes/no questions and are really effective. I also recommend being familiar and having information ready for disordered eating resources, right? So if you acknowledge that somebody has disordered eating or is struggling, that you can send them somewhere. And in particular, NEDA, the National Eating Disorder Association, is a really good resource. So they have online screenings for everyone, can tell you if they think that you meet criteria for an eating disorder or not. And then even further beyond that, it can connect you with local resources. So folks in the community who are NEDA certified, and then more broadly. And then for researchers, which I think is kind of probably more the folks in the room, I included this QR code. It is for the EDQ, which is a kind of the gold standard in eating disorder research in terms of measuring eating disorder risk. It is 28 questions, which I know is not ideal, but they do have a 12-question version and a 7-question version. And I recommend using this because it really is important for long-term follow-up. In particular for intermittent fasting, we see that when folks lose weight, at least temporarily, we see decreases in eating disorder risk and body dissatisfaction. But as we know, body dissatisfaction, weight gain, all of those things tend to come on later on in follow-up. So as folks gain weight back, because we know that weight loss is not particularly effective long term, as folks gain weight back, their body dissatisfaction increases and their eating disorder risk increases. So we want to make sure that we're not just measuring eating disorder risk pre-post, but we're measuring it long term so that we can really see how these things and our interventions impact eating disorder risk long term. We also want to make sure that we're not just measuring kind of our standard eating disorder behaviors in terms of restriction and fasting. We want to make sure that we're using more of a gender inclusive term. In particular, intermittent fasting is very common among folks trying to gain muscularity, and the EDQ and many eating disorder questionnaires do not assess disordered eating behaviors specifically for muscular orientation. So things like steroid use or protein powder use, etc. We want to make sure we're using kind of a more inclusive measure. And we want to make sure that we're measuring cognitions, not just behaviors, because as I said, cognitions can kind of rear their head long before cognitions do. That's all I have for you. Thank you. Jesse, anything else?
Nope. All right, I'm good.
Great, thank you very much. That is a really important topic, and I used to work in bariatric surgery for many years, and so I feel like I have seen this in action. There are many things that are good for many people, but are very harmful for other people. And so I think fasting very much fits into that group. You go ahead and stop sharing your screen so we can see everybody's faces for the panel discussion. Please.
I'm trying. Hold on.
Okay. Well, while you're doing that, I'm going to go ahead and think about the first question here. There seems to be a bit of people want more details. What can they have when they're fasting? What can't they have? Water is okay. Typically coffee, tea are okay, but how about coffee with stevia or coffee with cream? Where do we draw that line? Who wants to take that one?
I can take that one. We have generally do the following. So we generally say if there's appreciable calories that it would be off limits during the fasting period. So for us, a squeeze of lemon is totally fine. Chewing gum is fine. Diet soda is fine, though certainly not our first choice. We would say coffee with cream is not allowable. And we would draw the line there. We have done it either way in terms of allowing coffee outside the window versus not. It really just depends on the study protocol. I understand the research community is split on whether coffee should be in the eating window or not. We've worried a little bit less about it. And then in general, most of our studies were trying to isolate the effects of intermittent fasting independent of diet quality. So we generally tell people keep doing what you're doing. And so we've generally counseled folks to eat, just keep carrying on their usual habits. And then I will say, because the eating disorders presentation by Dr. Hahn was so provocative. One of the things we've done that I think has been beneficial for folks is that we generally tell them not to skip meals, which I feel like could be an important strategy for reducing the risk of developing eating disorders. So we just counsel them to eat the same meals, but in a shorter time frame. So
yeah, I would add
that
that
helping. Yeah.
That also aligns with the circadian data, right? It's about having that rhythm of foods, and the random skipping can do almost as much harm.
Yeah, exactly. I start worrying about that. And I also worry a little bit about these 1 meal a day approaches as well, where people try to eat everything 1 meal a day. And I think even though we don't have good data on this yet to say either way, but I worry that those also might impair protein synthesis.
Yes, I also worry about that very much so. And we have Dr. Kogan joining us, who's a geriatrician, who I'm going to guess also has some concerns about that. Do you want to weigh in?
Yeah, so I think when we're looking into a more older population, I think the risks are even higher, right? I think all of the studies that have been done, very few have looked at people over 65. I think, Courtney, I was keeping very close attention to your presentation because you presented so many studies. I think there was one that specifically looked to the older adults, but it was a small study. But I think the risk of sarcopenia, muscle loss in that group is even higher. And thus, I think we need a lot more data to say sort of what is safe, what is not safe. Generally, my position is probably 14 hours during the nighttime. Every night is safe enough, as long as you don't cut the calories. But going beyond that, I would be hesitant to recommend to a lot of people. Now, of course, there's a difference between 65 and 65, right? Somebody at 65 could look like 45, and somebody at 65 could look like at 95. We see all this. So it's not to be averaged here. It's really more of a general statement, I think.
So you mentioned sarcopenia. There's a question here. Could the increase in function in senior patients who practice TRA be a function of improvement in their sarcopenia, which has a strong metabolic component and consequently improves and the improved reduced insulin?
I think this has more to do with often sarcopenia is linked not just to sugar, but also to inflammation. Right? So if we know that we can control inflammatory response with the TRA, that or at least be part of the treatment for the inflammatory response, then that's probably going to be beneficial.
Absolutely. So similarly speaking, we got a question here from Dr. Brad Moore with the Lifestyle Medicine Clinic here at GW. How does lean muscle loss compare across the different styles of fasting? Are they pretty comparable, or do we see a difference? Courtney, could you answer that one?
Sure. In general, a lot of these studies find no negative effects on lean mass, but I suspect a lot of that's due to the sample sizes or the duration, and this is across a very wide variety of intermittent fasting approaches. I think it was James Betts recently in England, where I think he tried 36 hours of fasting on an alternate day basis. So I think it was roughly about ADF. And I believe they did see negative effects on lean mass within 3 weeks. Now, I don't think they used the most sophisticated methods of assessment where you can even tease out, how much of the effect is due to changes in total body water versus actual changes in, I guess, the non-water component of lean mass. So they didn't go that far with sort of these four I forget the exact word, but like these more sophisticated ways of parsing body composition. But I do think that is cause for concern. And I do worry about some of these more extreme forms of intermittent fasting in the long term, where you are having these 24-hour only fast. And I will say, I mean, I know we're supposed to talk only about science and not about anecdote, but there are a number of intermittent fasting influencers who said, hey, look, I did take the fasting too far and I lost too much lean mass with these extended 2 to 3 day fasts, or frequently doing, 1 day a week fasting.
Want to put a shameless plug to our geriatric team. We're actually doing a study on sarcopenia using a brand new method, which is different from standard. So if anybody's interested, I think I'll put the link. I'm not a PI, but a good close colleague in geriatrics is a co-PI. So it's a collaboration between geriatrics and engineering at GW. So if anybody interested, you'll get a free testing. I don't know if
There you go.
You may even get paid, but I'll put the
Excellent. Thank you for sharing that. Another thought that popped into my head too is, and you're hearing a lot about this with the GLP-1s, is when people lose weight, they lose lean body mass. It's by design, right? They were carrying this weight around and now they don't need to carry that weight around. So there is going to be some sort of shrinking. So even if there is a reduction, the question is that a harmful reduction or not? And there is definitely a level at which it's not harmful, and there's a level at which it is. And so you need these sophisticated tools to really be able to answer that. And I haven't seen any study that has looked at that. All right. So another question we have here about is the effect of TRE on hypothyroidism. For instance, any reason why someone on levothyroxine should not do TRE? Anyone want to take that one? Not my area.
There's not a lot of data on it. I think there's maybe only one secondary analysis of a clinical trial, and I believe there was no change in the thyroid hormones that were measured in that trial. Maybe there's two, but there's not a lot of great data. I don't have any particular concerns about it, but I'm also not an MD.
Right. We do have two MDs on the call, but I don't know if this is either Dr. Heyman or Dr. Kogan's bailiwick.
I haven't seen anything, and I have a lot of people on intermittent fasting. I would say, I don't know, Andy, if you've seen anything, I haven't seen any real effects. You know, I think you do have to be careful with adrenal system, I would presume, you know, but again, you know, we do this in such a carefully observed and controlled settings with monitoring that probably not seeing.
I've only heard conjecture that, you know, prolonged avoidance of carbohydrates, you know, potentially can affect T3 levels, but I haven't seen it, honestly, in my practice, quite frankly. You know, I think the benefits far outweigh the risks.
Especially if you're being monitored like they are in your clinic.
Correct. Mhm.
Right. Yeah, so I think that comes down to a lot of these questions. It's going to be how intense are you monitoring them, and are you working with them very closely? Then you can be a little bit more aggressive with these things. And hopefully you're also monitoring them for eating disorders so that they are not developing those. Let's go down to, there was actually a lot of questions about the eating disorder section. So I'm going to pick some of the big ones, and then maybe Dr. Hahn, you can answer some of them independently. What research has been conducted to identify at what point the risk factors become riskier as opposed to being reversible?
That's a great question. There hasn't been any research, to my knowledge, looking at kind of identifying what is a maintenance factor in particular, versus a risk factor for eating disorders in this population. So I think it's just acknowledging that folks who are going to be recommended intermittent fasting are baseline high eating disorder risk. And we just have to acknowledge that and screen for that and continue to rescreen, right, for eating disorders. And at this point, we really can't say what is a maintenance factor, what is rescreening, or causing eating disorders to be worse in this population, etc. I think it's just kind of the research isn't there yet, unfortunately.
Unfortunately, I think that's going to be true for a number of these questions because it is an emerging area. One of the points that I think maybe is not clear is exactly what do we mean when we talk about this eating window, right? Is 6 better than 8? Is 8 better than 10, 12? Where do we draw these lines or lines? And then do they have to be consecutive? I also got a question on that. Like, can you skip breakfast and dinner but have lunch? Or can vice versa? Can you skip breakfast and dinner or can you skip lunch but have breakfast and dinner? Is that enough to have some fasting benefit?
Yeah, I will answer from the eating disorder perspective in terms of what is high risk and what we consider problematic. And what we consider problematic is that feeling of deprivation, right? So being hungry and not eating, or being hungry and saying like, I'm not going to eat because I want to lose weight. That's really kind of the problematic sticking point for us rather than a specific amount of hours. We do consider like 8 or more waking hours as fasting and disordered eating, but because I think we can all agree that somebody who is awake for 8 or more hours is going to be hungry. And disordered eating, folks who experience disordered eating oftentimes have dysregulated hunger and fullness cues, in particular, folks who engage in intermittent fasting. They're used to prolonging their hunger, right? They may be hungry in the morning, but they're not eating breakfast, they're not eating lunch, they might not eat until dinner or what have you. So they're used to prolonging that. And we can't necessarily rely on their hunger and fullness cues in the same way that we can in a quote unquote healthy individual. So dysregulation and kind of saying, even though I feel this, I'm gonna ignore it.
Yeah, I love that. I'm all about listening to your body. I've heard that all the time, but you make a valid point that your body may not be able to tell you if you are,
And I'll
you know,
jump in
not
and
listening to it.
answer. I'll jump in
Go
and
ahead.
answer the second half of your question there about the skipping meals. And that goes back to Dr. Peterson, and she showed that nice metabolic graph of the different fuels in the body. And I think that really answers a lot of that question of the metabolic side. So if you go 6 versus 12 hours, there is a difference metabolically in the fuels available and what your body is trying to do. Is it trying to make its own glucose, for example? And is it turning on signaling mechanisms that do that, that turn on autophagy that we heard about earlier and stuff like that? So there is a benefit to going longer, and the research has shown that to an extent. I think that's what Dr. Peterson kind of strongly recommended, that 14 to 20 hours. If you break that fast by skipping breakfast, eat lunch, skip dinner, or whatever variation you want, then that's kind of disrupting that normal feel of that body. It's restoring exogenous glucose and fuels to your body and changing the signaling in your body. So when you do a fast, it's really that time of the fast is really where a lot of the benefit comes from metabolically. So there is probably some research, Dr. Peterson can jump in and tell me if some little micro fasts are good. And they probably are from a calorie restriction side, but not necessarily from a metabolic fasting side.
Good distinction. I can anchor that a little bit further. So I'll say the data on 10-hour eating windows is more mixed. We don't see a consistent trend with body weight. I mean, when you look at these meta-analyses, they pull them over windows up to 10 hours long. So net effect is, yes, it reduces body weight. But if you look at the literature in it with 10-hour windows, it's about half of studies find a benefit and half don't. I recently saw a study that compared 10-hour versus 8-hour, and they found a larger weight loss benefit, which is perhaps no surprise with the 8-hour window. I will say, in my experience, I have run experiments with 6-hour windows. We find that once we get to 6 hours, patients actually don't really like going much lower than 6 hours because they feel really stuffed. And actually, once the eating window gets that small, they find it more the actual eating portion of the day to be more challenging from the fasting portion of the day, because it feels a bit to them like they're eating Thanksgiving dinner every day, because they're just compressing all their eating to a short time period. So I don't generally recommend less than 6 hours. That would be kind of my limit, or at least in the absence of more information that it's safe from both a metabolic perspective as well from an ED or eating disorder perspective as well. So for most people, if this is appropriate to you, I recommend the 6 to 10 hour range. I recommend start at 10 hours, then work your way down if comfortable. If not, stay where you are.
I provide similar advice. I typically say aim for 10, particularly when I talk to seniors. I don't push them much beyond 10. But another group that could be of interest, there's a question here about the evidence or data on perimenopausal and postmenopausal women and women with genetic predisposition for breast cancer. Those are two questions, but I think we could just talk sort of generally about women's health and how fasting may affect women differently. Anyone want to take that one?
I can take it from a eating disorder
Great.
perspective. So as I mentioned in that bimodal curve, that is a peak period for disordered eating, in particular because women's bodies are deviating during this period from what is considered culturally attractive, right? Women are experiencing a hormonal shift that is really positioning them to have adipose tissue directed around their midsection, which is not what we consider conventionally attractive. And then also just more generally, their metabolism slows down, right? So women in this period are really trying to fight their biology to try to lose weight. And it's also a period where we see increases in type 2 diabetes, hypertension, all of these types of things where intermittent fasting might be recommended biologically for good reason. So I would say from IF eating disorder perspective, it is biologically makes sense, but it is very much socioculturally driven in terms of if we did not have these ideals, if we did not have these unrealistic beauty expectations of women and also of men, but particularly women, we would not be seeing these high rates of doing anything that women can to meet these unrealistic beauty expectations, including IF for those that IF is not recommended for.
Great, thank you. That was very helpful.
I can chime in too. There was a study that was just published in January in Nature Medicine in about 200 people. And one of the main goals of the study was to see if there were any major differences between men and women. And they didn't find any, which is interesting because in the animal literature, there are a large enough number of differences between male and female rodents that are intermittently fasted, but I'm not sure we're seeing any large effects in people, but who knows? We'll see. I mean, it's still a young field.
Yeah, I agree. And one of the things that pops into my mind when I hear it is similar concerns to seniors and people just generally. Are they getting sufficient nutrition? That would be my chief concern. And if they're, working with an RD or a CNS and making sure they're getting enough nutrition, then I'd be less concerned about that. But it's always going to be a problem with IF is, are you getting enough nutrition? There was a question here about people who are having trouble keeping weight on. And there are many metabolic benefits to this, but if you are someone who's naturally lean or maybe even too lean, is this a technique you should avoid?
I can probably speak a little bit to that since I deal with this all the time in clinical settings. So the way I sort of suggested the two things. So first you start with under the 14 hours, like 12 hours, and then you very slowly progress it. And you also always take some extra protein early with the first meal. I'm not calling it breakfast, just the first meal of the day. And then if the weight loss begins or starts, then sometimes I would just say, yeah, you have to stop. I mean, this is especially in people who are to begin with more metabolically fast. Sometimes I would even assess some laboratory tests. So there's a way to look at the Krebs cycle. And if I see that the Krebs cycle seems to be fine and there's not a lot of glycemic issue, they just want to lose, they don't want to lose weight. They're doing this, for example, as part of our brain program. So we give them these parameters and we monitor them weekly, their weight. And if we start seeing loss, we start getting worried and we either try to modify it. And if we can, then they just get off of it. This happens. And we're not necessarily monitoring the muscle mass, we're just monitoring total weight, and we're making sure it doesn't drop below, certain BMI.
I don't have any experience in this area, but I will say on the broader picture, I don't believe intermittent fasting is for everyone, despite what it might seem like. I do believe there's something in there in regards to meal timing for everyone. So for instance, simple rules like don't eat 2 hours before bedtime or 3 hours before bedtime, I think can still be really helpful for folks. And we find there are plenty of folks that are in occupations that don't allow them to do time restricted eating, or at least not so easily. It makes it much more challenging. And in those cases, there are other options too. I mean, to the best of our knowledge, there are benefits from eating earlier in the day, at least in terms of blood pressure and glycemic control. And so I'll often tell our patients who, can't do intermittent fasting, like, hey, you can still, if I were to give you another simple rule to follow, it would be make dinner the lightest meal of the day and make breakfast and or lunch the largest meals of the day. So just some thoughts there about, I think in the long term, we do need to customize this for individuals. And we have some data that'll be coming out soon that we find people are so interesting and varied in how often they want to do intermittent fasting. So even in our studies, we'll report a mean number of 6 days a week, but we find there's some folks who feel like they want to do 7. And when we say, oh, well, you really can do 6 and take a break day a week, they say, no, I don't want the break day. I'll fall off the wagon. And they feel very
Mhm.
strongly about that. And then we have other folks who are like, I can do this if I only have to do it 4 or 5 days a week. And that's my limit, 5 days a week. And so I think it's important to really understand that we need to kind of meet people where they are in their life circumstances. And there's definitely no one size fits all.
Yeah, and I would
we
add, I think that's one of the benefits of the fasting mimicking diet is you only have to do it 5 days a month.
Exactly. Exactly.
we found in our mold and Lyme population that about 80% of them, based on their transcriptomics, they're in aerobic glycolysis. And it's not a great strategy in the early phases of treatment to put them on a fasting regimen. It's not until we clean up their environment, reduce inflammation, and as they shift into oxidative phosphorylation, then they can sort of sustain the fasting state and burn energy appropriately. But what we find, if they don't do that, they crash, and we run into trouble. So I tell them overall it's a good idea, but I have to sometimes hold them back from wanting to go into the fasting state a little early in the treatment process. It's a unique population, but it's not an uncommon one, at least in our field of integrative. And so I would caution practitioners who might be seeing mold and Lyme patients, be a little ginger with that group in particular, because they can really get into trouble if they try fasting and they fall into that kind of push crash phenomenon.
And I would generalize it to anybody who's really toxic, probably should be super careful. Like, should just really not start with that. Like, you should really figure out why you're toxic, get your mitochondrial health a little bit better before you sort of jump on this, because we see complications quite frequently. Crash is a right term. It's exactly what happens. They crash. They just get much worse for a period of time until you make them support them a bit more.
Yeah.
I think I have the perfect follow-up question for this from one of our integrated medicine students, actually. How do you best advise healthcare professionals to practice harm reduction techniques if they have someone who's not a good candidate for IF but wants to do IF? Like, they're going to do it. How do you help them reduce the harm?
I'd monitor them a lot more. If I know they're going to do it anyway, and I want to keep them, and I want to have that relationship, I would probably just be very aggressive with monitoring.
Yeah.
Testing.
Sort of to Misha's end, I mean, I would say some of the kind of vital signs of toxicity, you'd want their anion gap to be less than 15. You'd want their urine pH to be above 6.3 or so. You don't want them too acidic, because if they're really acidic, they're going to run into trouble. And so there are kind of like a couple poor man versions of looking at that issue of acidity, and and therefore as a proxy toxicity. So I always measure their anion gap, and I'll do a urine pH. And so these are just kind of rules of thumb. This is, let me be clear, it's not evidence-based per se, although there's data around these subjects. But if you're looking for kind of a guide, because one of the things we all know is that there's sort of this bimodal distribution in our practice of medicine. You get a group that's relatively well and want to optimize, and as Misha knows, we also get the group that is unbelievably sick and unbelievably complicated, and you want to be incredibly careful with these people, because as he said, if they're toxic, if you push them, they're going to crash. So always make sure that they've narrowed their anion gap, their urine pH is not too acidic, and then I say, okay, maybe we can do some of these other things. Just kind of pearls of wisdom and rules of thumb. That's all.
And I'll go back to the broader question that was in the chat. I think was really trying to dig down to like the risks of your patients, and the risks was really general, right? And I think, if you take those risks and compare the eating disorder with the actual risks we're talking about with acidity and pH and lactic acid production and that crash that they could have, there's two very different types of risks there. So it brings me back to, You could ask someone, oh, do you exercise? And they will say, yes, I do. And you're like, great, you're healthy and it's a healthy person. But then it turns out they exercise for 7 hours a day at this compulsive level. Well, that's a whole different level of risk now. And it changes your mindset in relation to that patient's now risks, right? So with eating disorders, there's that risk. There's that risk that they could tell you, I'm going to do this behavior. And the more you learn, especially with your patient in front of you, then that has to change your recommendations to that patient and what things you need to look out for if you're still going to go forward with any type of prescription, whether that is IF or not.
Perfect. Thank you. So we are running short on time, so I'm going to go around and give last words to everyone. I'm going to go in the order of presentation. So Courtney, you are first.
I think my best last words were the ones about customizing this to the individual. So I think I'll just double down on this. So I think we are clearly seeing benefits for intermittent fasting for losing body weight, for improving cardiometabolic health, though perhaps not lipids, I would say, or at least not with intermittent fasting. And we're also starting to see evidence that it may slow the aging process. And certainly in animal studies, I didn't present this, we now have 4 animal studies suggesting that time-restricted eating increases lifespan. So I think the data is really exciting. The question is, how do we prescribe this safely and to what patients and for what conditions? And I think we found that adherence depends so much on behavioral circumstances. It's not, folks who have an early versus late chronotype or anything like that. So I think it's really important to get to know people and what they're willing to do. And if there are any risks, say if they're on an anti-hyperglycemic medication, there are increased risks for those medications. If there are risks for eating disorders or other sort of factors that are important, and then kind of work with the patient to customize the plan.
Great. Thank you, Jesse.
Yeah, so I think I just go back to the general principles of nutrition. That's kind of a theme we talked about, is that intermittent fasting is a strong tool, but we need to treat it as a lifestyle change, not necessarily just a tool that people can use it and then stop using it, use it and stop using it. I think it's much more of a long-term tool that if we as a society change our eating habits and structures around the principles of intermittent fasting, where it's okay to go longer periods of time without eating. We don't have to be surrounded by food. Our lives don't have to be built around a clock. It's noon, it's time to eat, that kind of stuff. And if we just learn to listen to our bodies, go longer periods of not eating, I think that can eliminate a lot of risk with a lot of health benefits.
I did skip myself, and I'm just going to reiterate what you said. That's fantastic. I'm with you. And Samantha, I'll give you the last words.
I think mine are just words of precaution, right? I think with every tool that we have in nutrition, I am an advocate of looking at potential harms, making sure that we are not just kind of giving blanket statements and saying this is a beneficial tool for all, but thinking about for whom this might be harmful. In particular for IF, the folks that we are giving this advice to are folks who are at high risk for eating disorders and disordered eating. So thinking about this from a more holistic perspective in terms of how are we advocating for IF, and is this actually, potentially harmful for this individual? Just thinking about it more holistically in general.
Well, we are all about that in integrative medicine. So we are out of time. This was a fantastic afternoon. I really enjoyed it. Thank you all so much for joining us. We really appreciate your expertise and your time.
Thank you.
Until next year, everyone.
Mhm.